Testing, 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 testing. Testing, 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 test, test, test.
Good morning. Could we get everyone to take a seat, please? Uh, Good morning, and welcome to Savannah, and welcome to this Western Boundary Current Continental Shelf Interactions Workshop. It's really a pleasure to finally see you all here together. I am sorry that more of you who are listening in online could not be here as well, but happy to have you. What I want to do, oops, what I want to do first is to acknowledge some of the people and groups that have made this possible. Oh yeah, please, yeah, thank you. Um, and of course, by the people who make this possible, I mean the people that gave us money. Okay, beginning with the National Science Foundation, who thought enough of this idea now 4 years ago to give us seed money to make it happen, and then last year gave us even more money to make sure that it happened. I want to thank the provost office of the University of Georgia, who gave us a whole bunch of money to bring in students. And in fact, we have 11 different students from 7 different countries um, represented as a result of their generosity. And I want to also thank our own institute, because what they have done is tell me that they will pay my cost overruns rather than me writing the checks, and I do appreciate that. One of our major sponsors is Socorro, the Southeastern Coastal Observing Regional Association. It's part of the National IUs, and they are responsible for funding and administering lots of coastal ocean observing activities in the Southeast, mostly gliders and coastal radars. We have some money from Seabird Scientific. Um, they're one of the premier makers of ocean sensors, and we have really a museum of seabird sensors in our at Skidaway, and dating back really to the late '70s, and I think they all still work, which is a real kind of testament to their quality. We have money from Teledyne Marine. Paul Devine is here from Teledyne today. Teledyne makes everything, but the Slocum gliders, RDI workforce, ADCPs. He's sponsoring the coffee hour. So once you wake up, give him a good thanks for doing that. Um, and anything, since Teledyne makes everything, really, anything you've ever put in the ocean probably has Teledyne components. And Helsel Nest Techniques, the makers who wear radar, have given us some money for the poster session. Uh, None of them could be here, but we have a poster from one of their people here today. We've been running Wear Our Radars here out of Skidaway for 17 years. They've been great partners with us for all that time. And a shout out to Joana Gonzalez from Mexico. She's going to do some social media outreach for us with her site. So if she wants to take your picture, please smile for her and give her a good quote. I'm going to try quickly to go through how we're going to run this. Okay, today we have in the morning our keynote speakers. All afternoon is free. Go out in the town, grab some colleagues, find a cafe table, find a bench in the park. Uh, Maybe not a great day to sit out by the pool, but meet with your colleagues and talk science. We'll be back here at 4 o'clock for a first poster session with refreshments, 4 to 5:30. Dinner break between 5:30 and 7, and then another set of shorter presentations from the community this evening. And tomorrow's the real work of this workshop. We're going to have it's the same setup: morning sessions, evening sessions, afternoon free with the poster session. Um, three thematic sessions in the morning, three in the evening. They will be upstairs on the second floor. Um, I would say there's a lot of overlap between these. These are not unique. These sessions, these themes sort of fell out of our discussions back in 2020 at our virtual workshop. You know, if you walk into cross-shelf exchange and it's kind of full, move over to scaling. It's ultimately kind of the same. You're talking about the same thing, right? So I regard these as these categories tomorrow as prompts. They're not commands. They're just a way of organizing, beginning to organize your thoughts. Go in there, talk about what you know about these topics, talk about what you don't know, more importantly, and talk about what it is that's keeping you from knowing what you don't know. And then also maybe, you know, trying to think about these systems in a comparative way. Is there a way that we can productively think about how these behave the same, and if they behave differently, what is it that makes them behave differently? By all means, mostly, let's not review what we know, but think about what we don't know and how we can move forward. Okay, and those sessions will be sort of facilitated by the people I call our workshop principals. That's Lavinia Ruin and Ian Sothens from the University of New South Wales, Federico Brandini and Ilson Silveira from Sao Paulo, Deja Wang from the Chinese Academy of Sciences, Eileen Hoffman from Old Dominion University, and Dana Savage from Skidaway. Unfortunately, our other three, Tarang Amon, Marjorie Krug, and Sydney Guo, could not make it to this meeting in person. And then on Wednesday, what we're going to do is what I'm calling our consolidation. So we'll all meet back here. Our principals will really report out on what we were talking about in the Tuesday sessions, and then we'll have sort of a breakout of the whole, just an open discussion of everything we've heard, every other point that we didn't manage to make on Tuesday, and hopefully really come to a general understanding of what everyone has done over the previous 2 days. Short lunch break, and then whoever doesn't have to catch a plane or catch some Z's can come back here and try to wrangle all those thoughts into a written document. The reward for that is we're having a post-workshop social in town at 5 o'clock at a speakeasy bar off of Broad Street. We'll get directions to that later. And then beyond that, I don't want the momentum to disperse when we all disperse. I'll take those written reports. I do have to submit a report to the funding agencies, but I'm hoping that somehow we as a group can then maybe write a review article, get that out into the public, maybe arrange for a journal special issue. I haven't gotten very far on that project, but some way to consolidate all our research and feature it together sometime in the future. And then probably most important, the connections that you make today, keep them moving. Start planning some research collaborations, maybe some cross-system comparisons. I'd really like this to be a catalyst for new research down the line. And the last thing I want to do before I get this thing really started is tell you why we are here in Savannah and why it's not someone else somewhere else that's doing that. And we are here because of these four people. And Skidaway back in the late 70s, Jack Blant, Larry Atkinson, Jim Yoder, and Gus Paffenhofer were here. Gus is still here. And they really worked out for the first time how the Gulf Stream and the frontal eddies interacted with the shelf to allow nutrients to be delivered to the shelf and feed the shelf ecosystem. Okay, and something like 90 to 95% of all the nutrients that are feeding our shelf here in the southeast come from these Gulf Stream, hidden Gulf Stream upwells. But more than those scientific achievements, which we have done a poor job of following up on here, it's how they went about it. And it was a very interdisciplinary, egalitarian team that did that work. And their way of doing science really imprinted on my institute how we should be doing science. And that their influence on us lasted a good 30 years after they and all dispersed and that work was over. And I want to make this workshop a tribute to their efforts and their influence on us, and I think really on some of you too who have followed up on this work in other systems. And with that, I would like to invite Ian Suthers up to actually begin this program.
Thank you, Bill, and thank you, Skidaway. Thank you, everyone, for coming a long way and for planning a long time. 2020, we first discussed this, and things have changed. Papers have been published, people have changed jobs, we've moved on, but it's great to see you all, and what is really a very brave endeavor, one that is almost overwhelming, which, as I led up to the preparation of this talk, the first thing that I realized was to remind us all of the sensitivity, the relationship that we have with our Western boundary currents. These population estimates, 5% of the US live cheek by jowl with the Loop, Florida, Gulf Stream system. These are fairly conservative ones related to the Sea Around Us project that looks at the actual fisheries landings from that particular coastline. But you see 26%, and again, a fairly generous error for the purpose of illustration to show the actual interaction with the shelf. 69% of South Africans, 36% of Australians between Brisbane to Melbourne, and nearly 90% of the Japanese population are cheek by jowl with a major warm river that runs beside them with all that moist air and driving all those characteristics, the upwelling that Bill mentioned. These also are having dramatic impacts upon our ecosystems, and I wanted to, in this talk today, was to emphasize the ecosystem outcomes of Western boundary currents rather than the physical processes. And this is the basis of a paper from my colleague Adriana Vergas, who showed that all the Western boundary currents are influencing the demise of kelp, the magnificent forests of underwater macroalgae, and are being decimated by the invasion of tropical herbivores. These herbivorous fishes are transported in the currents and consume and remove a lot of the kelps along the temperate coasts. And these temperate coasts are immensely valuable for ecotourism, for fisheries. The east coast of Australia alone is worth approximately a quarter of the marine economy, which includes oil, gas, and shipping. So I was thinking on how I could rationalize this, and overlooking the view of the Sydney Institute of Marine Science. Some of you, if you do, sitting here in the office on the lower right, those of you who land in Sydney would be there. Those arrows, the airport in the background, our University of New South Wales is in the background. But I wondered how to align all these 5 Western boundary currents onto a common template for something that we can raise this morning. So taking those 5 workshop themes that Bill has presented to us, you can see the consistent theme of the ecosystems, common ecosystem characteristics, and I'll address these in my talk this morning, looking at latitudinal gradients in fish communities, some common species that using growth and mortality that integrate the spatial and temporal variability of physical oceanography, and some fisheries outcomes. But I want to return to what I proposed back in 2020 that is now just recently published about how frontal scale eddy dynamics may unite Western boundary current ecosystems. And if you look from the Sydney Institute of Marine Science out through the Sydney Heads into the Pacific and towards the United States, we're reminded that the continental shelves occupy only 7% of the ocean surface, and yet they support more than 90% of the fisheries landings. And those are in the vicinity of all the major ports and harbors that take delivery of those fisheries landings. So we really need to understand what are the processes that drive that ecosystem, that support the fisheries, that feed those communities, especially in the decades to come. So it is indeed the fine scale features, ironically, rather than looking at the geography and the separation points and the latitude and the drivers, it may be the fine scale features that unite us. I was hoping that there would be more similarities that unite us than divide us. And I think it's through the fine scale features that we can do this. So my aims are today to describe some of these, one mechanism, one conceptual model of a modified ocean triad that was recently just come out earlier this year about these three processes that frontal eddies achieve by creating an offshore nursery ground for larval fishes. And then to the second stage of my talk will be to compare some of these ecosystem characteristics, looking at zooplankton and latitudinal fish gradients, some growth and mortality of some common fishes, same species that occur globally, and to look at larval fishes in western boundary current systems. On a personal note, and this is old news, and I apologize, some of you have seen this slide before, but it was in my career, one of those inspirational moments. We were steaming back from Lord Howe Island after looking at effects of the Tasman front around those islands, and had to meet the pilots off Sydney at 7 a.m. And we saw this wobble, this billow, this frontal eddy begin to form. And David Griffin, who many of you know, has set up ocean current, so I can show you some of these magnificent images of the East Australian current, said, you know, Ian, no one has really sampled a cyclonic eddy. And for me, as a biologist, that raised my eyebrows. And so I went up to the bridge around 10 to midnight, and there, fortunately, was the third mate on the dog watch, happened to be the son of the chief scientist. And I said, could we please have a few hours sampling in this area? And he said, not a problem. Steered the vessel, and we did 3 replicates at 2 sites inside this frontal eddy and outside. And you can almost see how the eddy is entraining that ocean color here on the left side of the image here, entraining the chlorophyll into this feature. And so outside the eddy on the way home, yes, there was typical assemblage of zooplankton and a few myctophid fishes, but inside the eddy, it was extraordinary, replicated over different scales within inside the eddy. Not only were they more abundant, but sardine, scad, mackerel, and of course the ubiquitous oceanic myctophids, but they were larger. So implying that they were enjoying improved survival. And interestingly, we can discuss this later, the growth rates were not significantly different to outside it, and there's a reason for that. In fact, they were enjoying less mortality. But also there were nutritious salps and little small euphorsids. It was indeed an offshore nursery ground, and we had the TV trucks down to meet the vessel back in those days. So then I got back into a bit of history, the work that Bill referred to, and the original studies in the Florida current. Lee, 75, examined spin-off eddies from this wave-like meander. These things are ageostrophic. They're just driven by the tangential forcing of the current beside it on the oceanic side. They are nonlinear. They are stochastic. They're difficult to forecast and predict, but they're there and they're regular. They are ephemeral. Physical oceanographers looking at the heat transport will think, are they really important? Well, we now know from studies in the Agulhas that indeed they do have huge physical oceanographic characteristics, but they're just that sweet spot, the little Goldilocks just right. 3 weeks duration, they can grow out and recruit. So they're ephemeral, but a few weeks larval stage. They can grow and entrain more and more, and then become geostrophic, can raise the isotherms in an upwelling favorable system, and are regularly are adjacent to the continental shelf. So Andy Bakun is a physical oceanographer at the Rosenstiel lab in Miami, and he wrote in his book to describe these things as an oceanographic triad. A triad as in the essential elements of a musical chord, just those 3 notes that give you a nice tone. And his conceptual 3-stage model was that these systems to work need to be enriched in some way to concentrate the larvae into a patch, and that they are retained and not just dispersed out into the adjacent open ocean. And the triad of evidence is evident in a series of papers by other authors in Mediterranean, Benguela, Humboldt, Eastern and Western Australia, which then Bakun later adapted in 2006 for the ocean eddies. And I wish to then go through this right now about how they can entrain the distinctive shelf community by entraining this thin green ribbon of preconditioned water. And we can see this, and I'll be showing you a number of examples where the inner shelf water is enriched, both with phytoplankton and zooplankton, due to a beautiful, interesting array of upwelling processes. They sustain that larval community because it tends to be upwelling favorable, but it also has very interesting dynamics when that when during spin up and spin down of that frontal eddy. And it retains it because that eddy rolls along the edge and is transported at 1/10 of the rate that it would have been if it would have been in the boundary current itself. So we came up with this little conceptual model. There you go, the 3 stages to entrain, sustain, and retain up to 14 to 21 days. And there's the thin green ribbon driven by such interesting processes such as friction by the bottom shelf, a reverse equine flow that allows deep water to intrude across the shelf, northeasterly winds, an equine flow that then completes the uplift into an upwelling and is manifest at the surface, at least, with enriched chlorophyll as well as fertilized by rivers. The entrainment of this thin green ribbon and the cross-shelf flows are a key component of the offshore nursery concept. The formation is equally physically fascinating, and there are a number of metrics, conditions that some people agree with and disagree with, that all in part of the mix would be suggestion, the occurrence of an opposing wind, the bathymetric characteristics of topography, a headland, or even a canyon. The studies on canyons are something else that I'd like to raise in the workshop the next few days. And definitely freshwater outflow through estuarine flows will also initiate eddy formation. And there's the interesting aspects that have recently been shown in the Florida current eddy, where these eddies, where they spin up and then spin down, either spin up, the isotherms dome, and spin down, the core relaxes, but there's upwelling around the sides, and then it can be nurtured again as the eddy gets squeezed, maybe further downstream against the shelf, spun up by the Florida eddy, and it uplifts again. And so that nurturing and that fluctuation is a wonderful aspect that I hadn't really appreciated until we started to write this. So we've explored these things to start off with East Australian current. Myself, my colleagues, Meninia Rowan amongst them, we've explored this system over a number of voyages. And with my grey hair, I've been blessed with 10 voyages on Australia's oceanographic research vessels. One of the best ones was a study we did with, it's published by Everdale and with some of Meninia's team, where a model recreation of a particular eddy just over 14 days, a little bubble, little billow began off the central coast of New South Wales. Sydney is just down here where my pointer is. And then just a few days later, this had now swelled out, and we could see that these particles that had been labeled across the grid were entrained into this frontal eddy. And then as it formed just 3 days later, it had pulled in, drawn in water from over 4 degrees of latitude into this eddy. And there it was now fully formed, and it lasted for about 3 weeks. It was only about 40 kilometers in diameter by the time that it collapsed. And on another occasion, earlier we found another frontal eddy, and you can see a series of them in SST here in the central panel, and on the right in Modus color. And we sampled the southern one and crisscrossed it with both this old-fashioned device with my LED optical plankton counter, doing toyos from the surface down to 150 meters deep. And we found doming of the salt and temperature within this eddy. We saw patches of fluorescence of the entrainment at depth down around about 40 meters deep. The isotherms had been, certainly isotherms, not so much in density lines, were lifted up by 30 meters from the surrounding. And we could see, even in real time on the plankton counter, that it was thick with material going through that counter. And as it turned out, with our toad nets, it was full of, you guessed it, juvenile salps, Thalia democratica, and little small euphosa shrimps, the early life stages, which are just bite-sized for juvenile fishes. A third example, in June 2015, we sampled an old cyclonic eddy, and then we saw during this massive structure that was nearly 80 kilometers across, and we only partially did it. We saw this feature begin to appear, and I made the call. We had to rush down and sample it, and glad that we did. And so we trisected both of those with nets and with a plankton counter. And what was interesting is that first thing came out from on this new vessel. It's stable. We got microscopes. We can sort at sea. We could see already in the frontal eddy there was a lot of sardines and small fish inside there. So we knew onto something hot using this multiple open closing net and surface nets. And to describe physically this, because these entrained fish are growing, they're also dying. And yet we have this ubiquitous background of the lanternfishes, the myctophids. We could have picked any other deep water oceanic fish, but we like myctophids because there's nearly 300 species. They've got spawning, one of those species has spawning all year round. So they're always there and they're constantly recruiting into the eddy. But these coastally spawn fish, which are all commercially important and really raise the blood temperature of recreational fishing, are these brim and snapper, little mado and sweet, which feed on the planktivorous, the zooplankton, mullets, goatfishes, carangids, and morwongs. And these were our coastally spawned fishes. And we made a ratio, and we took the square root of it just to stabilize the ratio, a bit like a temperature salinity plot. So this is the ratio, the square root of the coastally spawned fish in numbers per 100 cubic meters against the myctophids along the horizontal axis, numbers per 100 cubic meters. And there in the old eddy, we sampled at 3 depths of surface, 0 to 50 and 50 to 100. You can see that the northern coastal eddy had modest numbers. Clearly, myctophids dominated the catch. And then further out, we saw actually even more myctophids with a few coastally spawned fishes. But as you can anticipate, this frontal eddy was full of coastally spawned fish. And surprisingly, the source water up against the coast, where we saw the eddy actually being born, was dominated by myctophids. And we were banging our heads saying, how could this be? But we realized that in the movement of this eddy offshore, the East Australian current had flooded the shelf with the oceanic family. Within this frontal eddy, we also saw that it had entrained some estuarine water. It had a distinctively low surface water salinity signature. So we knew that even physically that we had this coastal water inside of frontal eddy. And this was the feature article that showed you this pictorially of this entrainment of this enriched coastal water and was put into the frontal eddy and the doming of the isotherms during a spin-up. And it was Meninia who reminded me to emphasize that this warm filament is a key characteristic of the frontal eddy formation. But what about were they sustained? What was the actual outcome? And one of the key parts of the ocean triad idea is that is this feature too ephemeral for predator populations to grow up and within it and start to attack those larval fish? What do the larval fish, do they have an advantage? And there were 3 dot points that we could put towards this was really that they were, that the water's already preconditioned with eggs and larvae. So they're constantly spawning on the hope that a frontal eddy comes by, that the real predators, the larger krill, would still take a little bit behind them to reach that point. And so they just scoot through. And Bakun and other papers have described this as a loophole for their survival. What about retain? So my colleague, Ahmadine Schaefer, as part of Meninia's team, looked at the results of the WERA radar of central northern New South Wales coast, published back in 2017. And there's an SST image with the HF radar superimposed over a beautiful cyclonic eddy. And beneath it, she has the corresponding chlorophyll image and the altimetry. You can see how that doesn't resolve the frontal eddy. And there are also surface divergent contours there. And what she found is that these eddies form irregularly, but about weekly. That's roughly about 50 per year. That their advection is less than the East Australian current, which is retained. And their eddy behavior is really interesting. That they go through the dynamics of spin up and spin down, we believe. And that some of them could be classified as rollers. They just roll along the inner edge of the East Australian current. Some get locked in behind a headland and persist there for up to months. And some what we call are scooters, where they zip along the front out into the Tasman Sea, perhaps a loss to the system and their cargo. But on other papers that we'll learn about more in this workshop, there is a pair of large recirculating eddies that seems to persist off the New South Wales coast that Meninia's team have discovered, and it's a larval superhighway that can jet them back in towards the coast at characteristic locations. The Kuroshio is a similar system, and the key point is, for me, in the lower Kuroshio, I do apologize to those who like to work further south, is Cape Shionomisaki, which is a really interesting location where this forms a wave-like train of undulations of the Kuroshio front. Every 1 to 3 weeks, Kumura, these are large frontal eddies, up to over 100 to 400 kilometers in diameter. The Kuroshio is the most interesting of the western boundary currents, having the least latitudinal change. At this location, it's not incurring all those changes in the Coriolis parameter by going further north along lines of latitude. Perhaps this is one of the differences that I'll allude to later on. Kimura estimates about 36 of these occur per year, and that they produce an amazing little model, and showing by Lagrangian drifters and by falling water mass characteristics that these anchovy larvae are spawned here, entrained offshore into the Kuroshio front. They're sustained by the process that I described, and then the eddy then spins off and collapses further up into the bay as they recruit. And Okazaki refers to these frontal eddies, or freddies as we sometimes call them, as an oasis in the ocean. And they said that the key process to their fisheries was this process. Kasai said the entrainment process, which is probably caused by offshore movement of the Kuroshio, holds the key successive survival and recruitment of fish larvae in the Kuroshio system. The Agulhas is perhaps the most intense of the southern. It is the most intense of the southern hemisphere. Its volume is more than the sum of the EAC and the Brazil current. The Natal Bight here and the Agulhas Bank are key nursery and the ecosystem areas. Downstream of the Agulhas Bank are characteristic eddies that are retained there for some months. Their formation is well described by a number of recent papers, including glider deployments. And that's where they realize the importance of these for volume transports into the Indian Ocean. But the actual entrainment process and their significance for larval fish populations is not as well described. But nevertheless, particularly Roberts and his team have shown, this is over a year of CWEF's images showing you the high chlorophyll inside the Natal nursery area, which is equivalent to our Stockton Bight off the city of Newcastle in Australia, and equivalent to the Enshu-Shinada Sea of the Kuroshio. These are wonderful parallels that we should be exploring in the next few days. There is this dramatic, I'll just pause here, a dramatic paper published by Teskey et al. In Scientific Reports recently, showing you the KwaZulu-Natal sardine run, regarded as the largest, one of the most dramatic migrations on the planet, well shown by David Attenborough's series, where there are two stocks of these herring, these sardine, and that the cooler sardine will move around into the cooler upwelling areas and begin to migrate up from spawning, but the cooler stock may be trapped and be physiologically compromised by the warmer Agulhas current. And there we've seen these images of sharks and birds feeding on these concentrated shoals of sardine. They did acknowledge, though, that these shoals are only about 10% of the actual stock. But I now return to where the story began, to Lee et al., and it's the loop and Florida current and Gulf Stream. We need to consider these three currents in this western boundary current synthesis. It's in the loop current where all the processes of entrainment occur along the coral reefs of western Florida coast, and where you have a semi-permanent gyre that circulates. I've got to get my anti-clockwise circulation correct in the northern hemisphere here, off the Tortugas reefs and the Florida Keys. But as they're moving along the Florida coast, you can see this chain of frontal eddies, and let's zoom in here to show you in ocean color the high patch of chlorophyll A. What is perhaps most exciting is Sue Spinogle's work and Bob Cowan's, where they've used light traps along the reef to sample these fishes, and repeatedly over many months, and they see the pulse of settlement occurring with the passage of a cyclonic frontal eddy. Not only that, but most importantly of all, the keystone of the triad concept is that they showed that that the first demonstration that there was significantly higher growth of larval fishes in those frontal eddies than in the Florida current. And they did this by looking at the little ear bones, the little otoliths of these larval fishes captured inside the eddy. Their increment widths are larger, meaning they're growing faster because the otolith grows in proportion to the fish. And this is evident in the settled they also capture with their light traps. A slower growth pattern in the Florida current was never observed in the settled fish. Not only that, but the eddy, as you expect, was cooler, it was more saline, and it had more fluorescence. So what are some of these ecosystem metrics now? To move into the second stage here, how can we do this? And I hope the next few days we can look at the Motus phytoplankton biomass with my colleagues that helped me to integrate the Motus patterns for all the 5 western boundary currents. But all these systems have temperate rocky reefs, and based on kelp and zooplankton, all have sardines, which are a great species that we should use into comparison because it's their growth and mortality that integrates the otherwise spatial temporal variation that we all know that come with western boundary currents. Some of them do have this wonderful species that we call tailor. I think you call bluefish. Others call anchoveta or scad. It's a wonderful coastal predatory fish, and we can compare its growth and mortality, and I'll do that in the next few slides. In this paper, to look at the latitudinal gradients of fishes along the New South Wales coast, we used the citizen science program called Reef Life Survey, which has now gone global. So they have surveys in the Kuroshio, of Eastern Canada, in the US, of course, and all around Australia. And it's an extensive data series over more than 10 years. 163 species were boiled down into just 5 trophic groups, and we examined the latitudinal and seasonal variation, that trophic composition. And the bottom line is this, is that up north, off the coast of Brisbane and the southern Great Barrier Reef, it's dominated by these herbivores and also the omnivores. But you can see the zooplanktivorous fish begin to dominate the temperate latitudes where the East Australian current rumbles along the slope. And then in the southern waters, which is far less biomass of fish, you see the benthic invertebrates starting to dominate that ecosystem. Down here, they have to deal with strong seasonality in that zooplankton, where it's much more stable up in northern waters. But interestingly, on an annual average, those 2 zooplankton biomasses work out the same, whereas this is always enhanced over the long term. And then we then looked at this. The blue is for herbivorous fishes from published work in the Gulf Stream versus the green are the herbivorous fishes, the purple are the zooplanktivorous fishes. And you can see along the Gulf Stream how the similar pattern is there, moving from herbivory to zooplanktivory along the Gulf Stream edge. Same as in the Brazil current, from herbivory to zooplanktivory. And obviously in the East Australian current, we found up north there was a dominance of herbivores, which are the herbivores that are now tropicalizing, invading the southern temperate kelp-based habitats, and increasing biomass of planktivorous fishes. We use an optical plankton counter, either an LED or a laser system in recent years to quantify the zooplankton across those gradients of the East Australian current. And this is old data that was only recently published. And the red shows you on a logarithmic scale the amount of zooplankton biomass that we found. You can see the path of the toad body going down to 120 meters deep. You can see the upwelling that occurs that drives the nutrients, that feeds the zooplankton. And we can see that the inner shelf is not just a thin green ribbon, but it's a thin, well, brown ribbon of zooplankton. And then we looked at the international studies of cross-shelf gradients in zooplankton or from all around the world. We found 21 of these and looked at the and this graph here shows you the ratio of the inshore to the offshore. So the ratio of 1, where the similar biomasses or similar zooplankton conditions is shown with the red dotted line. For the abundance, there's always 10-fold, 20-fold more abundance of zooplankton. They are frequently smaller, that's why. The biomass was 1 to 3-fold greater in the inshore waters. And the slope of that zooplankton size spectrum, which is a measure of the mortality to growth ratio of the zooplankton, was always steeper in close to the shore, where there's lots of planktivory going on. And this is a conceptual model to show that the inshore waters were smaller, but more biomass. The offshore waters were less biomass, but larger particles. And we had a steeper slope of the zooplankton size spectrum, that is a higher mortality to growth ratio. For the Pomatomus, we examined the distribution of the species around the world. You can see populations in the Gulf Stream, on the Brazil current, in the Gullis, and the East Australian current. I don't know why we don't get a population in the Kuroshio. And you can look at their fork length to age, which is their growth rates, and you can see that the Gulf Stream, and let's just sum it up, cut the chase here in case I'm running out of time. The Gulf Stream has the fastest growth rate with the least mortality. We can infer the mortality rate from the age distributions, the catch curve. And we can, and also, whereas the East Australian current and also the Brazil current and the Lewin current suffer the higher rates of mortality. So the growth in the East Australian current is the slowest and mortality is the highest. Why is that? Well, we'll get that in just a moment, I hope. One final interesting quirk with is that variable transport East Australian current does also drive the fishery. So these little tailor, the same species, we found a new spawning area from our larval fish, a long-term observing. We saw the catch, so this black line here shows you the catch per unit effort in southern Australia off the coast of Victoria, and the transport was estimated from a particle tracking model that looks at the East Australian current, and that the appearance of these fish coincides more or less with a correlation coefficient of 0.46, showing that the transport of these small larvae is essential for the recruitment into the nearshore waters in southern Australia. Last but not least is the larval fish community, which integrates all those patches. And we've been surveying these now since 2014 and at 5 locations around Australia. But as far as our East Australian coast goes, we can see, for starters, that the diversity is much greater here off the north of Brisbane compared to off Sydney, compared to off Hobart. But even more interesting is that we're seeing a strong rise in diversity. These are the new fishes that are invading the temperate coast from up north and are changing our ecosystems due to the strengthening of all western boundary currents. And as a basis for tropicalization. So in conclusion then, I'd like to, I saw one of the abstracts spoke about the Kuroshio paradox, that is an oligotrophic current. And Sato 2019 spoke of the enormous production. So forgive me for this busy table here, but it's something for discussion. But this is using the large marine ecosystem numbering system that you can go from the Sea Around Us website. You can see that in terms of the volume transport, this is where the population data came from, the shelf area or the inner fishing area, the percentage of tropical reefs, the primary production, if you believe that, but we can discuss that. The recent landings, I took just the average, eyeballed average the past 10 years. We can download the data and be more precise with it. And you can see at the fish, because there's no effort data, no hours at sea, no boats, but the number of fish per square kilometer is tenfold greater in the Kuroshio. It absolutely is a paradox. I think it's to do with the wider shelf there. It's a very wide shelf just off where those frontalides form, and also the basis of salts and larvaceans that dominate the tropical ecosystems. So hopefully, I'd like to conclude I spoke today about fine scale features. A lot of this research on frontalides is by serendipity, sometimes by planned opportunity. These studies had different objectives. The Florida current was to examine the tropical reefs. The Japanese research was to look at the anchovy and the sardine industries. And we were also just out there looking to opportunistically. So we need to then bring into the fold the Agulhas and the Brazil current. I hope that this afternoon and tomorrow we can look at some common species and metrics to look at that ecosystem perspective, and particularly looking at some of the larval fish production and sardine production, which I didn't get onto. So thank you very much indeed for your time. I hope this got some questions, and I wish to acknowledge my patient colleagues, but especially NSF and the Skidaway for hosting this wonderful workshop. Thank you very much for your time.
Thank you, Ian. What I'd like to do is, if people have questions, and then we have plenty of time for questions, because I was quick and he was too, to come up to the mic rather than me chasing around the entire room. So please, if you have a question, come on up. Let me start. You mentioned that the estuarine outflows seem to affect eddy formation. Could you elaborate on that?
I might flick that question to Meninia, but when I spoke to um, Matt, Matt Archer, who's my co-author of that paper, he said absolutely, definitely, that the estuarine outflow affects that because of the different densities. It's biologically, it's because of the more saline oceanic water that is being forced has now got a pool of lower density water that has enabled it to be captured. So physical terms, I might ask Dana to help me as well. But I also say in the same breath that it is not well accepted that estuarine outflows do drive that, even though Matt Archer's term is absolutely definite. I believe that canyons and topography, in terms of opposing winds, Amandine Schaefer investigated that, and as I recall, that she could not find the effect of opposing winds. They weren't necessarily related to formation. But it's a really important point because from this meeting, I want to be able to have a system where we can get down to the scale of trying to forecast these stochastic events. I'll leave you with, thank you, you've just seeded a question for me, for the audience, is that can we use these frontal eddies as a way to improve coastal fisheries? This is a crazy idea to get, I know you say it amongst friends, to get a container load of fertilized eggs on a helicopter and place them into one of those frontal eddies and go and measure them and look at their enhanced survival. Rather than 99% mortality, we could have maybe 50% survival, and then we'd look for those genes in the harvested crop 3 or 4 years later. Thank you, yes.
My name is Ricardo Matano. Thank you for the beautiful, great talk. I'm pretty ignorant, but the question when you put all these fascinating pictures about the eddies is, these are all ephemeral. So is there any quantitative measure of how much these eddies interaction will affect local coastal ecosystem and fisheries? For example, consider that an ocean in which there are no eddies will be fisheries significantly different, or the other opposite. Due to climate change, it will double. How that will impact?
Yes, that's a great question. And some of my colleagues also said, so what? You know, these things are so ephemeral. They do not are not frequent enough for us to observe and quantify. Why should we even be asking this question? Why should we even be spending a workshop to look at them? My answer is this: that the study in the Agulhas was able to model that flow with and without frontal eddy formation, and they found that more than 40% of the Indian Ocean, of the leakage of water into the Indian Ocean, was facilitated by frontal eddies, 40% of the volume. So if we look at, say, one larval fish per cubic meter, that ends up, and especially if they're getting much greater survival in those habitats, then you can imagine the implications for having an ocean without frontal eddies. I cannot imagine that it would ever happen, but I think it's a great system, a great question now that a lot of these models that are now quite sophisticated, we should run them with and without frontal eddy dynamics. And I think that would really answer your question. So to my knowledge, and correct me if I'm wrong, Meninia, but no one has actually run a system with on our EAC with and without frontal eddy formation. And that would be to go and address that. But at least they've done it for the Agulhas. Are you aware, for example, Bill, have people done this for the Florida Current Gulf Stream system? No. It's probably a challenging task to run with and without. In the Agulhas, they also have run, some Japanese scientists have run the Agulhas with and without as the Durban eddy to look at the formation of that so-called Natal pulse. This is where a frontal eddy, or an eddy, a cyclonic eddy, is occasionally released. And you can see it spinning all the way down and entering into the Agulhas Bank ecosystem. And what is interesting is that they've run that model with and without the bathymetry to show that the Natal pulse still occurs, and it's driven more by just the Gauss physics rather than the topographic features. So running these models with and without these drivers would be a really great way to answer your question.
Okay.
Yes, sir.
Jia Yan from Wuzhou. So you talk about eddies locally formed by western boundary from the instabilities, but ocean is full of eddies, so a lot of eddies are formed to the east side of western boundary. Are those eddies as impactful as the locally formed eddy? Or so I'm kind of thinking about that. The western boundary current, I mean, when it's become unstable, is a source for the eddy formation due to fundamental instability or volcanic instability. But it could also be a shear, I mean, due to the large potential vorticity gradient, I mean, mainly due to the slope, small current slope, kind of block the eddies from the vast open oceans. Any thought about that?
Yes. Yes, I have thought about that. And indeed, Bakkun's hypothesis was always developed for offshore eddies. So thank you for your question. We also see on the ocean side of the East Australian current, there are also frontal eddies being formed. And we will see throughout the oceans, wherever there is shear, you will see small cyclonic features, but also anticyclonic features. They tend to be more cyclonic, but we do see these throughout the ocean. So the answer is absolutely. Along these really big oceanic eddies that last for years, you will find the same dynamics, edges, and especially the edge effects. There's two things, however, that make it not as a wonderful system as you might expect. Even though tuners do target those systems, they do spawn around the edges. A very powerful paper came out in, I think it was Nature Climate, that spoke about the biomass of big pelagics with inside and outside cyclonic and as well as versus anticyclonic eddies. But the differences that affect the coastal frontal eddies are number one, the entrainment of enriched water and of coastal spawning. So it's entrained. That's a really important step to actually bring that fertilized condition in and to give them the jump on the predator population that grows up. So in your offshore oceanic eddies, they will always have that ubiquitous predator population, especially big old eddies. And we even see it in our big cyclonic eddies that are easy to see on the chart, direct the ship, they sample it, and they're not as dramatic from a larval fish perspective as we would have hoped. And I think it's because of the predatory processes that have now caught up, and now it's just like, standard game of grow fast and die before you get eaten. But these frontal eddies have just got an unusual situation which they can capitalize on. So we do see these features. They are nutritious, and heck, lots of global pelagics use them very successfully, obviously, because they're still here today. But I think from the coastal ecosystems, where 90% of those landings come from, it's these frontal eddies that can really provide an additional benefit that we just haven't realized. We see typically 99% of all eggs and larvae die before they reach maturity. Within the first 24 hours, approximately 99% of eggs are gone. And this has been shown for a number of egg dispersion studies that look at the mixing and the survival. The studies at Scripps Oceanographic Institute show you the little cameras drifting along with the tentacle around the eggs. The mortality is outrageous. It's extraordinary. How can they get a way ahead? I think it's in these unusual ephemeral habitats. And that's what Bakkun spoke about with these sort of predator loopholes, he called them. He was a great one for inspiring, I wish he was here today. He's a great one for inspiring unusual thoughts. And some of my colleagues would just laugh and throw their hands up. But he was always thinking about what's going on, especially at the fine scale. So thank you for the question. Yes, it does occur on a grand scale, but it's the fine scale feature that really delivers the outcome of the fisheries.
Ian, we have a question online from Hitor Totsi. Do you think that the mortality of the fishes in these systems is related to turbulence that disturbs the This is the current structure, such as a powerful storm might do.
So the question, thank you for the question, much appreciated, and that thought. So the question pertains to how turbulence in the ocean, it's an old Ruben Lasker hypothesis, that the upper mixing stirring enables the shear at the fine scale, enables the larval fish to come in contact with a nauplius, with a larval prey item. And when these two coincide, they can have a satisfactory meal. And so it's that fine scale microturbulence was seen as a really important parameter to explain this variable survival. That's a difficult thing to measure. And so the field and the concept has, while it's there in part of the mix, and the issue of storms that acts to then dilute any layering of the phytoplankton, and therefore dilute that horizontal layering of the zooplankton, the nauplii, gets mixed up. And so the larvae therefore are dispersed in that system, and the storms referring to the question, and therefore they don't survive as well. So storms do have a huge impact, and indeed it's just that environment where you have a little bit of mixing, but not too much. And so, as they've shown in modeling studies, you need to have a dome-shaped relationship that gives you just a little bit, but not too much. But at the bottom end of the day, it's really hard to balance these two things. How do we do this objectively? Because they're growing and they're dying. We've got turbulence, we've got layering, we've got temperature. It does your head in. So recently, we've been looking at some of the underlying principles of that zooplankton biomass size spectrum. That is, it has a linear slope, and I can talk about this during the workshop, of abundance or biomass from the very small, the nauplius sizes, right through to krill. So it's a fairly constrained view of size spectra. Many of our colleagues have done this in the North Sea, everything from bacteria through to whales, and looked at various spatial scales. But just in the zooplankton size, it's a very interesting area. And at the outer, on the right-hand end of that distribution, are the larval fishes. If you go back to the mathematics underlying the movement of a growing population without investing in reproduction, the slope of that linear spectrum is the M to G ratio, the mortality to growth. That was put out in the Kendrick von Forster equation. It is the slope of the M to G ratio. So the mortality to growth ratio is absolutely fundamental to understanding not only zooplankton, but also the distribution abundance of larval fishes, because that subsumes all the other competing hypotheses: turbulence, match-mismatch hypothesis, a parental condition, all come out in the M to G ratio. How do we get the M to G ratio? Well, you don't have to now look at those ages of those larvae, measure the growth rates, measure the mortality rates. You'll be there until and for another 1,000 years. But from the size frequency distribution of those larvae, you get the M to G ratio. So I think this is now a really exciting way forward, that we can test the effect of these frontal eddies, these fine scale features, in a logistical, feasible way, is to measure them using any kind of shadow graph, or using your ISIS, the Ichthyoplankton Survey Instrument, or plowing through your samples, as good old grad students do, measuring a length distribution, converting to a biomass, and looking at the M to G ratio will assess the effect of storms, the effect of eddies, the effect of rainfall, discharge, nutrients, upwelling, seasonality, pollution. Yeah, hopefully I've sold you on the M to G ratio.
Hello, Ian. I really enjoyed that, and your enthusiasm shows through. So I'm learning about frontal eddies and the interaction of western boundary currents with biogeochemistry. I was wondering whether you think nutrient supply is also an important component here in terms of these kind of features being able to pull up nutrients from the nutrient stream, which in the Agulhas is 400 or 500 meters deep. And another question I had was, which may be more from Moninia, you were talking about how, measuring these things is really kind of serendipitous. And what do you think might be a role for the future SWOT, particularly in the East Australia current, where you seem to have, I think I remember you had like the most of these features. Did you say 100 per year or something? So that might be a place where you're most likely serendipitously, right, or otherwise. But I was wondering, what opportunities SWOT might be bringing to your area in particular for observing these features.
Great question. I'll start with the second one last. I've been aware of some of Minia's colleagues been looking at SWOT, and it's very exciting to get the resolution now, to see these things form. I think it will enable modelers to be able to forecast these features much better. When we can see, we're not looking at altimetry to the same degree, but we will be able to combine that altimetry, whatever you can see, of just a few centimeters at the most, with the location, be it and that'll be coast specific. So certainly on our coast, we do have large canyons. In fact, we've got a cruise next year to look at these canyons, and I will be nudging those oceanographers to help us see if they do at least initiate the formation. Sue Allen's work of Oregon was really a point in terms of the canyon. But I digress. So the SWOT will absolutely help us, and we need to forecast those so we can get much more directed studies, because we really are flying blind. A lot of these successful studies, certainly in the Kurashio and the Florida current, were based upon larger features that they could see. They went and could sample them, and I actually think they're a bit too far down the path of becoming a stable ocean eddy and had a predator population that was existing within there, but certainly that they were benefiting from the growth. Which leads me to your first question about the nutrients. So here's the thing, that nutrients at 20 degrees Celsius, you take a few hours to day to get those nutrients inside phytoplankton. They will respond within about 4 days to then have a doubling time at, say, 20 degrees Celsius, which then takes you up to about another 14 days, to, depending whether it's a salp or a crustacean or a copepod, to produce a change in biomass. So it's really, the nutrient supply is important to sustain it and for the longer term, but we really need to get that preconditioned water. We need to have the nutrients and the phytoplankton in that thin green inner ribbon entrained and going before this mechanism even can get off the ground, because it just takes too long. By the time we reach 3 weeks, there's predator populations have already caught up, and they're starting to enjoy this new abundance of plankton as well. So it's I've struggled with nutrients, and while we still go to sea and we record them religiously as we need to, the biogeochemistry has a lag in it that is almost equivalent to the larval stage itself. So nutrients are a tough line to follow, but I'd like to look at the nutrients and what's happening up against the coast that produces that load of phytoplankton, gets those adult copepods or the salps starting to do their thing, so that when they reach the eddy, it's all happening together. We've got to align. It's like a dance, We have to orchestrate everything to come together to make it to produce a successful year class.
Okay, thank you, Ian. I think at this point we'll have another break for additional coffee and get back to this at 10:30. Thank you.
That's test.
It's not pressing. No, no.
Can we get everyone back in here so we can get Okay, I'd like to introduce our next keynote speaker, Federico Brandini from the University of Sao Paulo, speaking on sources, dynamics, and fates of marine organic matter production in the Brazilian ecosystem.
Can you hear me? Yeah. Well, looking at all of you, I really envy English native speaking people like Jan, for instance. I do not promise you to be so eloquent in my talk as he was, but I'll try my best. So I'm Federico Brandini from the Oceanographic Institute of São Paulo University. And I think I'm going to talk about one of the least known western boundary systems among the other ones, which is the Brazilian western boundary system. This is the summary of my talk. I'm going to give you some the highlights about the biogeochemical provinces in Brazilian shelf, sources of nutrients, and the origin of organic matter in these places. I will give you some examples of coastal upwelling and shelf intrusions of the South Atlantic Central Water and its biological implications. And there is one issue which I think is becoming very important, at least in Brazil, worldwide, but particularly in Brazil, which is the trichodesmium blooms and the role of trichodesmium in the nitrogen cycle. And I will briefly talk about some experiments that we've done on vertical fluxes and the role of the shelf exporting material to the ocean regions, and finally some gaps of knowledge to be addressed. Well, not necessarily in this sequence, but let's see what's going on. So this is the general circulation, very schematic along the Brazilian coast. Here we have the South Equatorial system. When they arrive somehow more or less at 10 degrees of latitude south in the Brazilian coast, it splits into two branches, two main branches, the North Brazil current and the Brazil current, which is going to be the main focus of my talk here. This paper here of two shore and co-workers, unfortunately, it should appear a few seconds later, but it shows more or less the amount of eddies, cyclonic and anticyclonic eddies. This is 26 years of altimetric data showing how many of these eddies are there in these places, especially from these parts here, when the Brazil current starts to interact with topography and is shearing with shelf water as well. Some of them are quasi stationary, like the Victoria eddy, the Cape Santon eddy, and the Cape Fear eddy. And they have important role in the biogeochemistry, local biogeochemistry. And we talk about one of them a little bit. So Brazil occupied more or less half of the southern Atlantic southern hemisphere, from 5 north degrees to 38 latitude south. And there is a huge diversity of coastal biomes. I think it's important to talk about them because they produce organic matter and material which is run out to the shelf, and certainly they affect at least the inner shelf area, and make resistance. We have 1 to 2 million hectares of mangrove forests from north to south. Of course, the big ones near the Amazon, they are 30 meters high. Then, we have 3,000 kilometers of calcareous reefs, which is more or less a common characteristic in the Western boundary system. 20,000 hectares of seagrass beds, sand beaches. The longest sand beach in the world, Cassino Beach, is 200 kilometers. People use it as a road to go from Brazil and Uruguay a long time ago. Of course, now they have roads, but before they used the beaches to do that. We have coastal lagoons, for instance, the Patos Lagoons, which is considered the largest lagoons, I think, at least in the American continent. Large estuaries, as you know, the Amazon River contributes this 18% of the fluvial water to the global oceans. And we have also the influence of the La Plata River in our southernmost shelf. So these places here, driven suggested to split the shelf of Brazil into these 6 provinces. In the Amazon, North Amazon, and the southernmost shelf usually use detritus and phytoplankton are the main source of organic material because of the river flows. Then you have the northeast and the eastern shelf, which is more calcareous systems, the benthic calcareous, which is the most important reservoir of carbon and carbonate. And here in this area here, which is going to be the main focus of my presentation, the South Brazil bight, which is the analogous of the South Atlantic bight here. So phytoplankton is very important. So all these biogeochemical provinces were suggested by Castro et al. In this publication, one of the chapters of the collection, The Seas, about a long time ago. So I more or less base my presentation and rely on these chapters. Okay? So let me focus first in the South Brazil bight, which is going to be the main focus, which is my main affinity regarding biogeochemistry of shell. First, I would like to say that me and many of my colleagues in Brazil were very much inspired by scientists from this era. And I know that one of them is here, Gustav Papenhofer. This is one of the first papers that I read during my PhD. Look at what these people say. Phytoplankton blooms develop rapidly within nutrient-rich subsurface summer trees. New production is 50% or more of the total. This is something that I am addressing now in Brazil. And these people, they talk of these decades ago. The object of the research report here was to determine the effect of intruding equal Gulf Stream water on the chemistry and biology of the South Atlantic bight. Well, this became more or less the red line of my own research, also, and many other colleagues in the 80s and the 90s. Intrusions of nutrient-rich Gulf Stream water is the most significant source of nitrate to shelf waters. This is almost the same as we recognize the same in the Brazilian South Brazil bight. Intrusions are probably wind-related. In fact, they are also. So, the similarity between these two is so amazing that if you can see the regression that Ed Finsel and co-workers did long time ago of nitrate concentration in relation to temperature, you have this equation here. Well, I did the same in the South Brazil valley. You can see that coefficients are almost the same. The equation is almost the same. Well, these maps, they were built up based on my historical method, inspired by the works done here in the south of France. As you can see here, these are the summer situation. I'm going to show you two contrasting seasonal periods that I did. So I'm going to talk about historical information, and then I will show recent information to see how much they are consistent. So here's the salinity. Yeah, summer season is the rainy season, and that's why there is a lot of freshwater runoff for like everywhere, business as usual, make the coastal water more pressured. But this is important for the shearing of the Brazil current and formation also of cyclonic and meanders. Due to the Ekman transport that we have here is the northeast that calls this Ekman transport to the offshore. There is coastal, there is places with uplands, the coastal uplands is very localized. And the phytoplankton, in terms of chlorophyll, they more or less respond quickly to these intrusions and uplands. Of course, in the coast during summer, the intrusions go very nearby the coast, and the tidal steering that mix the water and the cold water signature, you can see it. By the way, this is not satellite. This is tracing paper with Nanking. This is not satellite. Now, they show you the way we did this kind of work. We didn't have sensors, fluorometers. We collect water with Nansen bottles at that time. So many of my colleagues of my generation know exactly what I'm talking about. So these are nutrients that you can see phosphate, which is very much respond to this upwellness in the core. This is the same map, the same breed, sampling breed, but during winter time. And during winter time, a very important signature that we detected at that time was the advection of the Rio de la Plata plume towards the south Brazil. You can see the salinity, lower salinity, tones of salinity and temperature. It's very clear, very clear. And these plumes bring us a lot of phytoplankton, mostly diatoms and dinoflagellates. But as you can see, phosphate response comes with this plume. This plume has an excess of phosphate, but not nitrate. Apparently, this nitrate has already been consumed in the Uruguay shelf, and when this plume arrives, it arrives with very low concentrations of nitrate. I mean, very totally different from the classical Redfield ratio, but with a lot of phosphate. But remember that the La Plata River drained a very complex hydrographic basin in South America, crossing many crop fields. When they use fertilizer, there's a lot of phosphorus in this fertilizer. So that's why perhaps phytoplankton is not able to consume all the phosphate that you can see there. But they will be important for other things I will show you later. Because this plume brings iron. I didn't measure iron, but I don't have to do it. We know that iron comes from continents and shelves. And phosphate. These are two elements very important for diazotrophy, for nitrogen fixing. You'll see later. This is the climatology based on these satellite information of sea surface temperature and surface chlorophyll, right? From 2003 and 2019, starting from here. And as you can see, the winter time, and this is the distance from the coast. As you can see, always winter time along this transfer passage, this is an example. We always have a lot of chlorophyll in the springs. And during summertime, you don't see chlorophyll, too much chlorophyll on the surface. Because as we will know, it is in the subsurface is intruding. So these are more or less the same database, but on the vertical plane. So during summer, if you look at the stresses here, you can see this self-vertical fusion bringing phosphate and lots of nitrates, yeah, which is also the main oceanic reservoir of nutrients to the soil. And this is the winter time. During the winter, you don't see, of course, the intrusions, they are always there. The only seasonal difference that they approximate to the coast or they go away from the coast, but they are always there. But during winter season, at least as far as I'm concerned, looking at historical information during winter time, the meandering and cyclonic formations of the Brazil current is more intense. And I detected at that time a very strong shelf break upwelling, probably very much associated with one of these meanderings of one of these eddies. We didn't have satellite at that time. This is one of them. So how important is these shelf break upwellings we detected? Of course, some of them helps the intrusion to penetrate more towards the coast. Now, these are more recent review about what we learned in these decades, inspired by the work of the Skidaway scientists. We have a mosaic in the South Brazil of different fronts. I would like to call attention first for these upwelling fronts of Cape Frill. Cape Frill became one of the most important research, oceanographic research, dealing with the interaction of physics and biology in Brazil in the 60s and in the 70s. So that's why it was so important. Of course, it's an anomalous phenomenon in western boundaries. It's very localized, and it's due to topography, to the wind field, and somehow to shelf break to meandering of the Brazil current, which helps to happen. But anyway, it became so important in the Brazilian oceanographic portfolio that they built up an institute, especially to study the upwelling of Cape Frill, which is still there and still there. But there are still people very interested in working the Cape Frill upwelling and the consequences in a larger scale. This is a paper of Khalil and co-workers in 2001, and where they model and they study. They are interested in studying the filaments, the frontal filaments derived from this upwelling, and how these filaments can be used as a gateway, a pathway of particulate material produced in the upwelling towards the, the, the outer shelf. So it's important for the cross-shelf transport of particulate material. So these filaments can even interact with some eddies of along the border of the Brazil currents. So another important front that we know, which is recurrent every year, is the subtropical shelf front, which was defined by an Argentinian scientist, Fiola et al. Long time ago. So this already mentioned for you what this front makes. It changed completely the biogeochemistry of the South Brazil, right shelf during winter. This is in a schematic way. There is also two other fronts. One of them is in the vertical plane, right? Is the surface aligned front, and the other one is the bottom parallel front. The surface aligned front defines the border between shelf waters and two tropical waters, and it was defined by our very good colleague, Professor Belmiro Castro, one of the greatest physical oceanographers in Brazil, which unfortunately passed away 4 years ago. And he was the one who defined these fronts. And the bottom thermal front, of course, in northern literature, they call tidal fronts, because the intrusion goes, the tide mix, and the front is retracted. But Belmiro called it bottom thermal front. But what is important also about them is that they divide, they split the shelf area, the shelf sections into three sections. The inner shelf is considered the area, the shallowmost area between the coast and the limits of the tidal bottom thermophone. The mid-shelf, which is between more or less between 50 meters and 100 meters isoband, is in between the two fronts, the bottom thermophone and the surface aligned fronts. And the outer shelf, yeah, is everything out of the surface aligned front is considered the outer. So this area is very complex, lots of fronts, lots of interactions, and the ecosystem and the cells, they respond very much. So there is a mosaic of different communities of phytoplankton that respond to these differences, and these are minor biogeochemical processes. Right? So here you can see that chlorophyll is really usually associated with the plume of La Plata River, especially diaphragm. This dinoflagellate here, this is a more specific community, Serraceum lineatum, which is an indicator of subantharctic water from the Uruguay and Argentina. They came together with the fuels. But usually diaphragms are very important to this. Coccolithos forests are species from warm water. I'm not talking about Gephyrocrasa or Coccolithos huxley. These are big tropical species. Of course, they are not inside the plume, they are out in the warmer water. And then, well, at that time when I counted, because I counted the cells, that's why I use glasses today, counting all the time. So, at that time, we didn't know what was Prochlorococcus or Synechococcus. We called them monads. It's a very fancy name, right? They were very, very small, but we used to enumerate them, to count them, and I put it here. And today, I almost know that they are ubiquitous, they are everywhere. This is probably Synechococcus cells, because now we see them in our recent work, we see exactly the same small cells. And here we have a population of Trichodesmium. As you know, Trichodesmium is a cyanobacteria, is a filamental cyanobacteria, considered one of the most important nitrogen fixers in the world. It was discovered in the 70s. Before discovering trichodesmium, people thought that nitrogen fixation happened only in crops of leguminous, in the roots, and then they found that no, the ocean fixes as much nitrogen annually than terrestrial crops. Yeah? And then, but they also said in the literature at that time that trichodesmium is rare in South Atlantic, because they used to say that the North Atlantic blooms of trichodesmium came from the dust from the Sahara. It's the Sahara desert dust that brought iron. Yeah? But this is not what happened here. So I put this picture in the shelf break in the northern part of the South Brazil. There is a lot of leaks, especially when the wind stops 2-3 days later. Oh, what happened? Trichodesmium comes out. Well, it's a mystery why they formed these blooms. We know mechanically why they formed these blooms, but why? Is there any evolutionary advantage to do that? We know that trichodesmium makes photosynthesis. They produce oxygen. And this oxygen is a poison to the enzyme called nitrogenase, the enzyme that fixes nitrogen. So how come they can produce something that is a poison for another thing? So there are some research, they find out, they discover there is some biochemical metabolic process that separates. They make photosynthesis at day and fix nitrogen at night. This is because they don't have the heterocyst cells, which is a cell that in freshwater cyanobacteria, diabotrophs, they protect the enzyme against oxygen. So these organisms, they do not have these cells. So how do they can protect themselves against oxygen? Well, we know they produce a lot of polysaccharides when they are here in the floating the surface. A lots of dissolved organic matter, and they attract a lot of heterotrophic bacteria. And these heterotrophic bacteria can consume the amount of oxygen inside these colonies. And when they do that, they cause episodically phalaноxic conditions sufficient to the nitrogenase to function. So perhaps they evolve like that. So they need to get nitrogen, especially in a tropical place where there is not too much nitrate available for them. We know that with turbulence they can go down there and take the nitrate. But perhaps this is one. I talked about this with one of the specialists, and he confirmed, yeah, it might be. This is a good idea. But I never proved this hypothesis that there is a kind of coevolution between trichodesm and heterotrophic bacteria. They produce the organic dissolved organic matter for the heterotrophic bacteria to consume oxygen and give them a good environment to fix nitrogen. And then they can do whatever they want to do with this nitrogen when they go down. This is a combination of these diamorphic cells, most trichodermal all over the world. As you can see, along the western boundary system in Brazil, they use data on cell occurrences, but they do not have too much data on the rate of nitrogen fixation. We never did this here. And also nitrogenase-based molecular genetic studies to conclude that the nitrogenase is functioning here. So this is something that we are starting to do now, not in time to speak with these people to put in the map, but this is an act of information that must be here. But the studies on these trichodesm is very old. Since 1963, a Japanese scientist described a bloom of these trichodesma in the northeast, where the shelf is very narrow. So there is, according to the wind conditions, especially the westerlies, they push these blooms towards the coast, even on the beach. And this produces a kind of, we call Tamandaré, which is the city there, where they detected this bloom, Tamandaré fever, because the irritation in the eyes, their muscle gets a little bit weak. So this was the first study they done. And then they stopped. They never did any more. So we are recovering. We are just making this study more. There is now a recent study about this made from people from the Rio Grande Institute, where they investigate the occurrence of trichodermal cells on the top of the device. And they found out that most of these surface slips is usually happen along the shelf edge. So the paper very much argue that the eddies brings iron and phosphorus and all for these people, so they can fix nitrogen. And well, as you can see here, most of them are in tropical waters in salinities above 46, and they use a very good regression, exponential regression between the cell density and temperature, showing that most of them concentrate above temperatures of 22 degrees, especially when you have the blooms in the surface, the temperature must be high. Well, it's easy to understand why it's high. They just go to the surface when the wind is weak. There is no wind at all, so there is more irradiation, so the temperature goes high. Doing like historical data also, I performed some photosynthesis lifetimes in the shelf with C14 methods, and I quite understand that I realized how much the Tmax, the photosynthesis rate at light saturation, is very much related to the amount of phosphorus. You can see here 0.3, 0.22, 0.34, 0.6. So the more phosphorus you have, the high is the photosynthesis, the maximum photosynthesis rate. And this is the compilation of all data that I got in my PhD, looking at the relative ratio in relation of temperature. As you can see, above 20 degrees in the tropical water, you have very, very high ratio, confirming that phosphorus is a problem. phosphorus is lacking, probably because they are demanding for the nitrogen fixation. I don't know. This is something that must be proved. This is just, you know, going back here. Just to keep studying my curiosity about the intrusions that was inspired by Skidaway scientists, I asked the Navy people to help me establish just one cross-shelf transect off Itajaí, Santa Catarina, one of the states in the south of Brazil. And I wanted to sample many samples. We sampled about 14 stations from the 20 meters isobar to 140 meters isobar. And we measured the wind here. And as you can see here, this is January. In January, the wind was very strong from the northeast. So now looking at the data that we got at this time, well, was a time that now I have a temperature sensor, a CTD, and a fluorescent sensor, and then allowed me to make this kind of map. So here, starting from November 2005 until June. As you can see, the isobars of 20 degrees more or less here. But during January, with the persistence of the northeast winds, you see how strong was this intrusion towards the coast. Right? And later in the season, the inner shell tidal stream makes the bottom tumble from the tidal front goes back. And here is an interesting phenomenon. This is, you can see this joint is the signature of the La Plata River in the inner shell. And below the La Plata River, which is very, not very dense, very pressure, you see the tropical water going underneath is an intrusion of tropical water invading the bottom system. What is interesting about this intrusion, the benthic ecologies are very interesting in this phenomenon because during summer season, which is supposed to be a warm season, the water is very cold in the bottom. And during the winter season, where the bottom should be cold, is warm because of penetration of tropical water to the bottom. And here in the right hand, you see the deep chlorophyll maximum layers that responds immediately. Of course, they start in the inner shell, they start from the bottom, and they more or less climb up the thermocline going offshore. So this is a very interesting result. So Atkins and co-workers, one of them, they say this. Diatoms generally dominate such intrusions. It is important to remind you that this chlorophyll maximum layer is a ubiquitous phenomenon, especially in temperate seas during summer and in tropical seas. But in oceanic areas, usually the formation of this deep chlorophyll maximum layer is very much associated with photoadaptation of cells. What cells? Well, cyanobacteria. It doesn't mean that the biomass is higher. It's just photoadaptation. They need more of the pigments just to get the less photons that are out there. But there is no new production because of that, no export production because of that. But in the shell, that's what I'm realizing in the south of the Brazil. This deep chlorophyll maximum layer is more formed by diatoms. And diatoms has ballast, right? So they export material. Of course, these diatoms, they also photoadapt. They need to do that. They are shade adapted hidden flora, like the Germans say. And what is interesting, what kind of species that I found? I also look at the microscope along there to see what are the species that are there. And one interesting that I found is there is a lot of Cosylodiscus wilesi. This is a gigantic diatom, which is typical from the Pacific Ocean and came with ballast waters to Brazil and invaded the estuaries and is starting to invade this deep chlorophyll maximum. I don't know how good they are for the copepods. They probably will increase the vertical fluxes to the benthic system, but we don't know exactly what is going on. And also this Alauposera granulata is also common. This is a freshwater species that probably comes with the La Plata plume. We don't know what a freshwater species is doing there. I was very curious, but I talked with a scientist from South Africa, and she told me that I found the same here. I found that. So we have to ask her what we do. So then I decided to integrate, make a trapezoidal integration of these chlorophyll stocks in the water column and arrive in these results here. In white, you see the amount of chlorophyll per meter square in the euphotic zone. In gray is in the dysphotic zone, and in black is in aphotic zone. As you can see, as the season progresses, you see more black here, right? It means that most of the stock is going down. So there is a flux, a vertical flux. And I also make a study of diversity of this species. Just look at here. You see, during this single here shows the seasonal period. And this one here, in freshwater triangle, is January, exactly when the wind is stronger, the northeast winds, and then the Ekman transport is the spring, and bring coastal diatoms towards the outer shelf as well. And I make some similarities, and I realized that exactly in January, 60% of similarity I found along with assemblage. You know? This makes me drive this following conclusion that there is a hydrodynamic loop of diatoms in this area, which is more or less like this. The number one, diatom cells in the coast are subjected to steering. So you find, if you don't see deep chlorophyll maxima, you find them in the surface, etc. During summer season, they are pushed away from the coast, reaching areas with less nutrients, and they become heavier because of poor metabolic conditions. So they start to sink. And when they sink, they found the picnocline, a nutrients rich area with still enough light. So they start to grow. So there is real new production here. When they grow a lot, there is a thickening of the deep chlorophyll maxima. And this compromise the light conditions of the other cells, so self-shading, right? So they sink down because of lacking of light, going to the bottom, waiting for the next year intrusion to go back to the coast. And well, now we are trying to assess the contribution of the deep chlorophyll maximus to primary production, something that these scientists from Speedway already did many decades ago. But now I have this fluorescent sensor. So we did many measurements with these fluorescent sensors. We detected many of these deep chlorophyll and deep instantaneous photosynthetic rates. We make some calculations and transform them in primary production in the euphotic zone. And just comparing it with other places, yeah, we have here, we found here that these are the results that we calculate in the South Divide in the past with C14, discrete sampling, and incubations. We found about 0.4 to 0.5 grams of carbon per meter square per day. Well, looking at the contribution of the deep chlorophyll maximum, we calculated 2 to 4 grams of carbon per meter square. I make all the further calculations about this looking at the fishery production. And we realized that, well, for this amount of fishery production, 0.4 to 0.5, if I multiply this for this dense geographic area, is not enough. The organic matter must come somewhere else. But at least if we consider the deep chlorophyll maximum, I think this solves somehow the problem. Well, now we have more recent work made now in January in the same breed, measuring lots of other biogeochemical, phytochemical, and biomolecular studies. And we found some interesting results which are very consistent with the results we found 30 years ago. So we have here the winter time with a very strong temperature and salinity signature of the La Plata plume. Well, these are euphortic zone and the ratio between euphortic zone and mixing zone, just to show you that light is not a problem there. And this is in summer season, so there is no plume, but there is some upwelling that I'll show you later. Well, these are nitrate ammonia is something that we measure, and we find out that not only silica and phosphorus, but also ammonia come with the plume. The plume comes with a lots of organic material, so ammonia is heavily alive. And, and this is in summer season. I do not have time to keep going on this, but there is something. This is also chlorophyll. I was expecting in winter more chlorophyll than I'm seeing here, but in summer it's more or less business as usual. Low chlorophyll concentration at surface because we are probably below surface is the deep chlorophyll maximum. Right? And this is something interesting. I took this index from Gruber and Sarmiento study, so they calculated this. Index, this N index. This N index, they say, is an indication of the surplus, of how much the NP ratio is deviated from the classical red P ratio. So if this any index is positive, it means that the excess of nitrate is coming from somewhere else, not from physical processes, and probably from nitrogen fixation. And as you can see here, most of this things, especially during winter time, is much higher here in the warm tropical water. And my argument is that the plume is bringing phosphorus and iron and favoring this nitrogen fixation. During the summer season, you see that not too much. And the silica index, you know, the silica and nitrate is absorbed in the same molecular ratio. So if you have the difference between them, if you have more silica in relation to phosphorus, it means that you have an area which favors the growth of diatoms. And if you have negative values, an area is not so favorable. And this is very consistent with what we expect to see. So you see in the plume during summer is very favorable for diatom growth. While during summer, diatom is not very good, except in the upper area of Cape Freedom, of course. Oops. Well, we are now starting this line. We are very delayed in vertical flux studies in Brazil, but we are starting now. So first, our first experiments, there are other experiments in Cape Freedom area with other scientists, or they already did. But in these shelf-rated or deep ocean areas, we wanted to know how much this shelf system contributes to carbon to the ocean basin. So we deployed two cellular tracks, one over the 900 meters isobars and another one over the 2000 meters isobars. And well, we found interesting things, of course, in the 900 meters isobars. The fluxes are huge, sometimes one order of magnitude than in the deep ocean. But there is a lot of contribution of lithogenic material, probably because advection of sediments from the slope that affected the results. So we still have to trust a little bit more on this thing. And in the 200 meters isobars, we have a lot of contribution of diatoms and coccolithophores. And these are more or less the nature of the biogenical particles. So we have diatoms, silica flagellates, and a lot of coccolithophores. Just to show you the sort of studies we are starting to do. And very interesting comparing to the South Atlantic Basin. As you can see, at 400 meters here, we calculated a yield of 34 milligrams per thousand of meters squared per day. And here, done by Biscay and Anderson, American scientists in the South Atlantic Basin, in the same isobar, we calculated 33, which is really similar. So, just to conclude, these are the scientific gaps that I would like to address with future investigations and in collaboration with one of you. of all of you. Intrusions of nutrient-rich South Atlantic Central America. How much do they contribute really to the net climate prediction and exploitation of organic matter in the middle and outer shelf section? Shelf breakup values associated with meandering of the Brazil plate. Do they explain the export of carbon in the adjacent percent layer? Coastal rock valley and Cape Verde, alongshore filaments they produce in France are trapped and transported offshore but mesoscale eddies. We must go with a little more detail and see how much they are important for the cross-shelf export of material. What is the contribution of cyclonic eddies? This is go with the question that Ricardo Matano made for you. We also have a curiosity to know how much is the contribution of cyclonic eddy for net primary production and carbon export in Brazilian shelves. How much atmospheric nitrogen fixation contributes for new carbon uptake in Brazilian shelves? So they put new nutrients in the water, especially in the oligotrophic system. This is very important. Iron and phosphate rich Amazon and La Plata regions, layers of perfume and nitrate depleted waters. We must address much better this issue and answer this question. What controls the seasonal pattern, if any, and the nature of particles exported to deeper layers in shelf and offshore? Thank you very much and ready for questions.
Thank you, Frederica. Do we have any questions from the audience? Anyone want to respond here?
Hello, Frederic. Thank you very much for the interesting talk. One of the questions that I think that this meeting in general should also address is what will be the impact of climate change on cross-shelf exchanges? Now, in the southwestern Atlantic, there is already evidence that because of the change in the winds, the Brazil-Malvinas confluence is moving farther south, which means warmer advection of waters along the shelf. The question is for you, and perhaps changes in the Rio de las Plata discharge. The question for you is, what do you think are the implications of these climate-related changes to the marine ecosystem on the Brazilian shelf?
Well, I have that there is. We can address this in two ways. For instance, if the climate change affects the precipitation regime, you can have La Plata plumes much more voluminous, and this can make some difference, bring more phosphate, more nitrate, etc. But on the other hand, all these processes that happen in the interaction of the Malvinas-Brazil conference, for instance, they will still be there, but probably they will be displaced. I think so. I cannot precise exactly in terms of absolute quantitative terms if what the ecosystem responds to these interactions in terms of production of organic material. They will magnify this production, or the production will be the same and just will shift position. But of course, what I'm thinking is that about the upwellings, for instance, if you have differences in pressure gradients because of climate change, you can increase wind forces, and therefore you can increase upwellings, and there you can have more production. They usually in the literature they say that the climate change, the warming, they can favor production because of they intensify the winds and hence they intensify the upwellings, or they can do the opposite. They stratify the water and they avoid and they limit nutrient fertilization to a 40 I really don't know exactly which answer would be the best one, but I think that we need further studies to address this. I hope that I more or less answer your question. Thank you for making them.
Hi, I'm Alex Sousa from Simbastav in Mexico, and I'm just having similar things in productivity. But we went and we can clearly separate what's the continental, or not clearly, we're trying to separate the continental impact from nutrients from the oceanic one. Can you do that? And can you see difference in the species that are more continental than deep ocean in your phytoplankton?
You mean in terms of global change? If the following up of the question,
Have your nutrients that you which one is more dominant? You have the upwelling part, or you have the continental part from Rio La Plata, and did this bring you different species from the oceanic part or the continental part?
I don't I don't know if we have enough historical data. I mean, just to define exactly. I mean, they have to modeling what is going on, but for sure, in phytoplankton studies, there is not a succession, there is a sequence. This sequencing happens because phytoplankton they answer very fast. It's likely change faster than 2 to 3 days. And as far as I see from my experience in the South Brazil biome, as you can see, there are uh, biological provinces which respond very much to this mosaic of different processes. But one thing that we must consider is about pollution, is about the anthropogenic effect. Of course, the human is putting a lot of things in the water that, at least in local scales, can change completely the diversity. You see, for instance, the diatoms that I found in the deep chlorophyll maximum, Coccinodiscus violesi, that came from the area of this gentleman here, is, is, is a species large and is occupying all the space, a space that otherwise other species would be there. So how much this will affect the flux? Because fluxes is very much associated with diversity, taxonomic diversity. That's why we're making these electronic investigations about our material also. So yeah, but I agree with you certainly. What we are doing with the sea, there is no more pristine air in the ocean. You can forget about it. You see mercury in tuna fishes in Pacific Ocean. You see DDT in Antarctic fishes from the 50s. Everywhere we are making a lot of bad things. So I think that first, at a local coastal scale, because the population grow a lot, demand uh, resources, and of course the coastal scale is much more vulnerable to this kind of changes. I hope that answers it.
Okay, thank you, Federico. I think we're going to have to move on. We can continue these conversations, obviously, for the rest of the week. We're going to pause for a couple of seconds till we get our next speaker, but thank you again. And while we're getting our next speaker, reminded again of something I forgot to tell everyone. The best Wi-Fi is DeSoto Wi-Fi.
Hello, hello. Okay. All right. So my title is The Problem of Scale in Assessing Physical Oceanography for Interdisciplinary Purposes, and something I've been thinking about for a while. Here it is. And I'm going to discuss it in the context of the South Atlantic Bight. So as you know, the South Atlantic Bight is on the southeast coast of the United States. Let's see if I can do this. Here we go. Extends from Cape Canaveral at about 29 degrees south up to Cape Hatteras at about 35 degrees north, not 29 degrees south, 29 to 35 degrees north, so about 700 kilometers. It's broad off Georgia and South Carolina, and it narrows to about 520 kilometers off Georgia, narrows to 50 kilometers at either end. It's shallow and flat. The average depth is only 27 meters, and our shelf break is at about 60 meters. So a majority of it is in the photic zone. We have distributed small rivers, but not a lot of freshwater input. The bottom is sandy with occasional exposed hard bottoms, and I'll direct you to Clark Alexander in the back of the room to get more information about that. And we have extensive wetlands on the shoreward side, which affect all manner of things. The Gulf Stream adjacent, as you know, exits the Florida Straits and runs along our coastline, along the slope, until it separates at Cape Hatteras. There's a feature here sort of in the middle of our coast called the Charleston Bump, which is represented by diverging 200 to 600 meter isobaths. And it actually deflects the Gulf Stream away from the slope for a short period until it rejoins the slope again off North Carolina. So we'll talk about that again in a minute. It continues to be unknown whether our coastline is a source or a sink for atmospheric carbon, and our own interests are in its potential to be a shelf pump to export organic carbon to the deep ocean. And we also don't know if that happens or what the magnitude might be. So I'm going to show you some sections here at this red line. Okay, so an outline. The fundamental process we are interested in the South Atlantic Bight is the Gulf Stream meander-induced eddy upwelling that spreads across the shelf. A little discussion of what that might mean to the carbon story. We have some large-scale convergences that suggest some export regions in the South Atlantic Bight, so I want to address those as potential avenues for exporting any organic carbon that might develop on the shelf. And then we're becoming more and more aware of the importance of smaller scale processes than we have historically been as oceanographers. And I'm going to talk about why that's an issue for physical oceanographers, and then discuss, I hope, some paths forward that might help us engage better with more cross-disciplinary issues. So the issue, you've seen this now several times already, is Gulf Stream meanders form off Florida. They travel north, and they grow and decay as they go along their way. They have eddies, frontal eddies that upwell cold, nutrient-rich water. Sometimes they entrain these warm streamers or filaments, and we've heard already this morning several reasons why we would be interested in those features. Our interest in the South Atlantic Bight, at least from my perspective, is not really in these filaments. It's in this cold water that is upwelled, and it's nutrient-rich. And this little schematic shows that in summer, when the winds are directed to the north and the stratification is vertical as opposed to horizontal, that cold water can spread across the entire shelf, and it's subsurface, so it's not visible in satellite imagery. Anything else I want to say? There's very strong temperature, nitrate correlation in that upwelled water. And so I've highlighted here 18 degrees. I'm not sure that the one that Federico showed wasn't for a little higher. The Atkinson paper maybe for a little farther north. But this is for the southern South Atlantic bight, and there's also a very strong relationship in the northern South Atlantic bight. It's just offset by a degree or two warmer. So just to emphasize, here's an 18-degree isotherm, and you can see that everything cooler is 5 micromolar or higher in nitrate. So it's a large number. It's a robust relationship. And so here's those sections that I had the red line, and I've highlighted the 18-degree isotherm here that's spread, and there's a little arrow pointing to it here. So it's, on the occasion of this particular intrusion, has spread all across the shelf and occupies a large portion of the lower water column. There's also very high nitrate associated with that onshore intrusion. And chlorophyll A in particular matter is elevated. All this is Pappenhofer and Lee, and of course a lot of this work, all of this work is based on the work of that group. Here is a map, a plan map, also from the shipboard sampling of the bottom layer extent of these plumes. And so once again, I've tried to identify the 18-degree isotherm, and that's a large quantity of 18-degree water. That's the sampling was not completed to the north because you can capture the northern end of it, but this extends over 150 kilometers. These are large intrusions, and likewise with elevated bottom nitrate and elevated bottom chlorophyll. So the long shelf extent is determined pretty much by what the lower layer circulation is. There can be reversals that move it back towards the south and the narrowing shelf, but we don't actually have a good sense of what the ultimate fate of these intrusions is. However, the diatoms can bloom and peak within about a week and utilize all the nitrogen that's well under the shelf in about 2 weeks. And there have been a couple of zooplankton evolution after that documented. One can be relatively small copepod species that can eat the smaller diatoms and leave the larger ones behind. And they can complete their life cycles and presumably feed fish eventually in about 4 weeks after the peak phytoplankton bloom. Another possibility is that doliolids or salps can develop, and their life cycles are different than copepods. They can double in hours or days. They can clear the water column at 40% to 120% daily. And so they are quite different objects. They can also respond quickly to depleted nutrients and diatoms and collapse, apparently sinking, and have been conjectured to provide a useful vector for shelf organic matter to get to the deep ocean, at least to the bottom. So I have got a list of references here, and I'm not a biologist, so I will defer questions on this topic. But the point being that these intrusions last on the shelf. They have been documented with shipborne surveys to last in excess of 4 weeks before something happens and they go away. But whatever happens to them is typically longer than our ship surveys have lasted. And so, is there a carbon pump on our shelf? We don't know. We don't actually know if it's a source or a sink. For atmospheric carbon, there's some dispute. There's recent modeling that suggests, from Najjar et al., Cahill et al., that suggests the shelf is a sink for atmospheric CO2 and a net exporter of organic carbon to the shelf. On the opposite side, that Kai et al., Yankee and Blanton have postulated that the shelf is a net CO2 source to the atmosphere. And so the reason we don't know is that it's a large area. We don't always know where they are, and we have not identified where they go. There's not enough data. There can be some complicated circulation. It's a difficult area to model with very large velocities at the shelf edge and an abrupt shelf break. That is a recipe for difficult modeling. And the meanders dominate nutrient import. I just put up this table from Yankee and Blanton And what I'm pointing to here is a 10 to the 10 in moles per year. The shelf edge upwelling dominates all other sources of nitrate and phosphate to the shelf by an order of magnitude, greater than groundwater input, river input, or atmospheric input. And so this is the process that supports new production on our shelf. And so my interests and that of Bill has been sort of, where does it go? Does it represent something that we really should be taking account of in the global carbon cycle? So that was all from shipboard work. There's also a way of assessing what it looks like elsewhere. Sorry, banging on my. So with shelf edge moorings, because of the strong temperature nitrate relationship, you can use shelf edge moorings of velocity and temperature to estimate cross-shelf fluxes of nitrogen and momentum. And so Lee et al. Did that. It's a very nice paper, Lee Atkinson and Lee Yoder and Atkinson, where they took those eddy moorings from a range of places along the shelf edge. And what they were able to do is associate regions of eddy decay with nitrogen input into the coastal ocean. And then they were also using Redfield ratios, estimated how much organic carbon that might support if it were all utilized at Redfield ratios. And so you come up with nice maps like this one. This is a winter-spring map. And this area on the North Florida, Georgia shelf is an area of eddy decay and nutrient input. Likewise, downstream off the North Carolina shelf is eddy decay and nutrient input. And then here in the middle, there's something called the Charleston Gyre, where I told you the bump deflects the Gulf Stream and it allows more space between the Gulf Stream and the slope and more upwelling. And so we'll talk about that a little more in a minute. Okay, I think that's all I wanted to say about that. You can do this for other seasons also, and I'll draw your attention especially to summer, where the carbon production represented by the gray here is extended into the shallower ocean. And then I'll also like to call your attention to the reversal in mean direction of the circulation in fall and winter, in fall in particular. So there are import regions in the south and north South Atlantic Bight. There's extended carbon production even near shore in the summer. And there can be a transport reversal in autumn. And we'll talk more about that deflection in a minute. So does anything get exported or does it just die in place or end up in place? Well, the sands underlying our South Atlantic Bight shelf are non-accumulating. They're classified as not accumulating organic matter over time. We don't know if the carbon gets respired and is exported from the coastal ocean, but there are some reasons to believe that the water that it grew in might be exported. So I'll talk briefly about large-scale convergences. At Cape Hatteras, there was a very nice 2-year mooring array that bracketed the separation of the Gulf Stream. And so from these, we were able to calculate along-shelf transports and along-shelf transport convergences. It's a very narrow shelf, so these 4 moorings are each at the shelf edge. Did a reasonably nice job of constraining what the along-shelf transports might be. And it turns out that the transports at Cape Hatteras are robustly convergent 90% of the time in the daily data. And so a lot of shelf water, both Mid-Atlantic Bight and South Atlantic Bight shelf water, goes offshore here. So SEEP1, SEEP2, Ocean Margins Project examined Mid-Atlantic Bight export. Not a lot has been done to examine South Atlantic Bight export. It's harder to tell from Gulf Stream water because it's salty and warm. But the along-shelf transports themselves correlate with winds, but the export correlated with Gulf Stream position variability. So that was a nice result. And Lujan, Harvey Syme have backed that up from some of our recent data with Peach in a more complete way and more recently. So Bill and I estimated what the South Atlantic Bight transport and export would be, and we came up with a number that was pretty similar to the export estimated for the mid-Atlantic bight shelf waters, which is maximum in late winter. This is in Sverdrup, so 0.12 off the minus means offshore, southward. And the South Atlantic bight is also about 0.12 maximum in summer. And this excludes any entrainment along the Gulf Stream on the outer shelf, which would be very hard to quantify. Also excludes the large volume of mid-Atlantic bight water exported in the Gulf front. Okay, so that export at Cape Hatteras correlates with Gulf Stream variability. In the southern South Atlantic bight, it looks like it might correlate more with wind. So what I'm showing here with the black line is a full transition. The black line is the near bed wave velocity. And so when the wind picks up, the wave picks up in the fall, the winds reverse and blow to the south. And what you can see is the red and the green are near bottom, and the green is near bottom, the red is near surface chlorophyll A, measured with optical instruments that we had on the observatory tower out there. And these have been calibrated to the best of our ability by Jim Nelson and Charles Robinson. And what you can see is that when the winds and waves come up, the chlorophyll value in the entire water column is elevated. And so our conjecture is that what's happening is that whatever is growing in these intrusions over the summer does accumulate in the sands temporarily, and then gets released in the fall with these energetic events. The wind is reversed, so it's going to the south. There we go. And so at a location here mid-shelf off the Georgia shelf, this is just a near surface and near bottom progressive vector diagram during one of these events. And what you can see is that, what am I doing? Maybe I should use this for the time being. So what you can see is here's a red dot, here's the red dot, so that's the starting picture. You can get in one of these events about 100 kilometers along shelf. And several of these events occur every summer. So it only takes a couple of, every fall. It only takes a couple of these to get yourself down here to where the shelf is quite narrow and export off shelf is required by continuity. So in this case, we seem to have another vector for off shelf export that correlates with the wind as opposed to off Cape Hatteras where it correlates with Gulf Stream variability. And then here's a very nice, here's yet a third possibility for export. So here's some very nice modeling results by Gula et al. In 2015. And what they show is that their model can capture the demonstrated bimodality of Gulf Stream position off just downstream of the Charleston bump. And so these little insets here are from Dewar and Bain, 1988. And what you can see is the model can result both the large deflection and the smaller deflection modes of the Gulf Stream. And so at each of these dots, what we have here are probability plots of what the sea surface height would be. And these three are all upstream of the Charleston bump, all south of the Charleston bump, and they don't really show any evidence of bimodality. These two are off South Carolina and the south end of Onslow Bay in North Carolina, and they very clearly show a bimodal character. So that's encouraging that you can do this with a model, and it helps validate that what we seem to have seen for years in the satellite imagery, and hopefully in the future with SWAT altimeter. And then by the time you get to this northernmost dot, that one off Onslow Bay, it has once again apparently returned to the shelf and is not participating in a bimodal deflection. So long time ago, as part of my master's work, I looked at some very nice 4-month mooring data, which is a very long time in those days, off of Tom Lee's mooring data for the Coastal Ocean Response Experiment. And I could demonstrate using these current meter moorings that the near shore and the small deflection and the large deflection resulted in a difference in shelf circulation with reduced southward flow with the more offshore displaced Gulf Stream. So that was, Savage did all that was Savage, Lee, Clanton, and Evans. I keep doing this. What am I doing? Okay, so here's the hypothesis from that paper that it illustrates how the more increased Gulf Stream, that hypothesized that the more offshore displaced Gulf Stream results in a reorganization of the surface height and the density structure in a way that then encourages more southward flow. So I've returned to that data recently and calculated transports from just those moorings. Maybe I'll back up one to point out that up at Cape Hatteras, I had 4 or 5 moorings in a 50-meter wide shelf, but I've done the same here with 3 to 5 moorings on a shelf that's twice that wide, and so maybe a little suspect. But when you calculate along shelf transports, what you find is, here's just a little schematic that shows both the nearshore deflected and the offshore deflected Gulf Stream. And so in red here are, when it's in its minor deflection mode, the transport at Myrtle Beach is 0.06 faradrips. At Charleston is also about 0.06 faradrips, which is consistent with the transports Bill and I calculated up near Hatteras for the same season. And I don't have decent data for Georgia, but I'll just conjecture that it's the same, 0.6. But for the offshore deflected Gulf Stream, the longshore transports are reduced dramatically. So that means about half off Myrtle Beach and way down to 0.01 off Charleston. So if the transports off Georgia and off Cape Hatteras don't change with Gulf Stream deflection, which we don't think they do, then that would imply some along-shelf convergences and off-shelf export. And so that's just kind of the idea. Now we have 2 more zones where we might be exporting organic carbon and no evidence to show that we actually are. Well, that's not quite true. So here's another piece of circumstantial evidence, which is a quite old map of the distribution of organic carbon by percent dry weight in superficial sediments. I've zoomed in on the South Atlantic Bight. And so the question is that, well, so you see the elevated regions up here near Cape Hatteras, here off where the Charleston Gyre is, and here in the southern end of the South Atlantic Bight, the 3 places where I've claimed we may have some export going on. But the question is any of this, is this generated in the deep ocean over the slope by open ocean upwelling, or is it actually exported from the shelf? And so that's a central question that we don't yet have an answer for. I will point out that up here, even at more elevated values, that's the depot center that SEEP 1, SEEP 2, and the Ocean Margins Project was designed to try to find the source for. And I think the end result from that was it wasn't Mid-Atlantic Bight export, the export of Mid-Atlantic Bight shelf water, because that community structure was composed of very small consumers and then expired quickly. And so it did not seem to indicate that that was going to be an effective vector. But yet here we have a whole lot of organic carbon up here also. So that's an open question. And here's just with the other maps I've shown you. So I feel kind of privileged to summarize this work, well, most of which is not mine. And I also feel privileged that and it's an important body of work on which I think the reputation of Skidaway has long resided. And I also feel lucky to have been associated with these people for my entire career. Jack Blanton and John Bain were my master's advisor and my PhD advisor. I think John's online, so hi, John. John and Tom Lee were on my master's committee. Larry Atkinson was just a tremendous mentor. Sorry. I thought I was going to carry my water up here. When I was a postdoc at Old Dominion, of course, towering figure that has inspired a lot of us. And since coming to Skidaway, Gus Papenhoffer has been extremely generous with his collegiality and expertise with both Bill and I. So thank you for that, Gus. So, but where have we gotten to since? This work is 30 to 40 years old, and Gus has continued to make progress. Adam's going to show you some interesting results also later on in the day. But we haven't gotten at this is what about carbon question? And that's because the intrusions are subsurface. It's hard to know even when they're there and when you should go sample them. It's hard to sample them with shipboard because you are not out there long enough in regional vessels. It's hard to put your moorings in the right place, and you really need real-time reporting observatory assets in the right place to know if it's even there. So they are a problem to measure, and they're large. They're long-lived. I think I kind of said that. The potential for carbon sequestration may depend on which category of consumer grows up in any given intrusion. And I don't think we have a good way of predicting that. We don't model the intrusion process well yet, although that's getting there, certainly. And we don't have any real good sense of what the interannual or even event-to-event variability is. So there are big question marks on our coast. And the pathways we've so far just hinted at. And in the meantime, we've begun to understand that the small-scale complications are maybe more important than we have been pretending they were. So as I said, I calculate along-shelf transports from extremely sparse cross-shelf moorings over just a 4-month period to make these claims about export pathways at the Charleston, downstream from the Charleston bund. So what about those small-scale processes? So I thought I'd just give a few samples. I think we can all come up with lots of examples of our own. I wanted to first show this. This is some more nice modeling work by Gula et al. This time 2016. And what I'm showing here is for a region off North Carolina and Onslow Bay, 3 different model resolutions. And so this is 1.5 kilometers, 500 kilometers, 150 kilometers, quite a fine mesh for the surface and for 200 meters. So the shelf is missing because our shelf is 60 meters deep at most. So, and what I just want to point out really quickly, and this is kinetic energy, so you can just think of it as a tracer for the purposes of this plot, is that every time you increase the resolution, you change the details of the surface circulation significantly. And so have we gotten to the true answer here yet? Well, that's, part of the modeling process is that's what they're driving at. How fine do you have to get to model appropriate processes? And then off the shelf break, the upwelling that's indicated by these patches is also changing. So likely how it spills across the shelf edge is also changing with resolution. So this is not a solved problem. This is a difficult process to model properly. And these are small scale issues leading to this result. I also wanted to talk about tides a little bit. So we've done at Skidaway a sequence of studies in a small marsh nearby, Groves Creek. This is my colleague Mal Heron, who came and helped me set up a radar, a very high resolution radar, to overlook the marsh and see if we could measure velocities in the intertidal area. Maybe here are some people collecting fluorescence samples because the purpose of this was to study how the tide moves through the marsh using a dye study. And so these were taken with a helicopter and just a subset of a really interesting set of pictures. But you can see the dye come in and then spread out through the marsh. And how does the tide actually behave in the marsh is a really interesting question. But I really wanted to show you these modeling results from Al Garrett. And I just thought I'd quickly page through them. So here's the simulated dye release. And you can see it comes in, it overspills its banks eventually, and off it goes in whatever direction it's going to go. And so by this point, probably depending on the wind also. And unlike on the shelf or in a tidal river, when you get to high tide, the water doesn't just gather itself and go back out the same way it came. It keeps going in the direction it's going even after high tide. until the water level falls far enough that it prevents it keeping going in that direction. And so it's complicated. Okay, so now finally we're going to gather ourselves and go back out. And then in the sequence, these marshes are kind of like a sponge. You have to rinse them over and over to get rid of all the dye. This is some nice work that Jack did with a treated water spill to prevent a nuclear plant some years ago. Here's our little marsh. It's about 2 kilometers across. There's a helicopter. Anyway, so what? Does this matter? Well, it does matter. So here's a nice modeling study by Brian Blanton that shows comparison between some ADCPs and 2 model results with and without an estuary, all the estuaries represented. And the quick answer is if you don't include the estuaries, you don't get tides right on the shelf. And so here's another depiction of that. This is amplitude and phase arrival of the tide. And so you can see that if you don't, this is a ratio of with and without an estuary. And so including the estuaries affects how well you can model the tides on the shelf. So that's kind of important. And getting model results correct for the entire marsh system is non-trivial. The Gross Creek Marsh is probably one of the best bathymetric, we have probably the best bathymetric map of that marsh as anywhere. So here's a little map that shows just this much of the Georgia coastline. It's an aerial picture of Google Map or something of the complexity of these marshes. But I wanted you to see the scale here. So here's the 10-kilometer scale. Remember, Gross Creek was only 2 kilometers wide, so it's about the size of this little bitty marsh right here. So getting that all right, modeling it correctly so you get the tides right on the shelf is non-trivial. Okay, so here's another small scale problem, shelf edge tide correlated eddies, which we've detected in the WERA radars we run here at Skidaway Institute. And so we'd see these little eddies spin up and shoot off to the southwest. So this is not a Gulf Stream meander-induced eddy, but it's a small-scale thing. We went looking for them with an artificial neural network, and what we found was they pop up at the shelf edge, and they pop up with tidal frequency. So when the tide sweeps clockwise past counter parallel to the Gulf Stream, these little eddies spin up fast. And then they shoot off to the southwest faster than any eddy could actually go. So it's just a disturbance of some sort. And then they dissipate within an hour or 2. So then they're gone. But we actually put some moorings in, did the same thing Lee did with temperature and an ADCP from the bottom. And it seems that it might be important and certainly more often than an eddy intrusion event. So maybe you need to get the tides right to get that process right, because it seems like they are transporting nutrients in the right direction here and maybe elsewhere along the shelf where the tides are right. So that was in a DRL paper. Another small scale problem, we all know that fronts support along front velocities. This happens to be the Hatteras front, which is a boundary between the Mid-Atlantic Bight Shelf water and the South Atlantic Bight Shelf water. This is for 4 days that I measured during the Finch project when the front was moving southward around Cape Hatteras and then began to retreat. And then we also towed an acrobat behind to get CTD information. And so for these 4 days, we have this one repeat section that I've shown with the red line. I hope you can see that. And so the acrobat data seems to show. So I should say that immediately before those 4 days, Hurricane Alex mixed the front surface to bottom. So you did have Mid-Atlantic Bight water resting on South Atlantic Bight water in a horizontal density gradient. It all mixed down. You still have a horizontal density gradient, and then it begins to relax. And so you've got, by a few days later, you've got a density gradient that's now not vertical anymore. Not the iso signals aren't vertical anymore. And what you see is some of this new temperature salinity values beginning to drift down vertically along these isopycnals. So that's interesting. That's an interesting mixing idea. Or is it horizontal? Well, I've got 4 repeat sections from 2 days, and those, so August 8th is on the right, August 9th is on the left, August 9th is on the right. And I guess the takeaway here is it's complicated. So from a couple snapshots like this, how do we determine how the mixing proceeded and what was really going on? And it matters in terms of the circulation along that front, which matters to the residence times of the shelf waters affected by that point. So, okay, that's interesting. One more example, Angarget found full depth laminar cells at 15 meters per depth. So we have also found them now on the Georgia shelf at 26 meters depth, which is almost the average depth of the entire shelf. And Yargo de Mello's got a poster back here that's going to show some data we have that appears to show them in a disorganized fashion, but full depth at 40 meters. 40 meters depth means almost our entire shelf can be affected by laminar circulations. And the point of those is that the very strong vertical velocities lift wave mobilized sediments out of the benthic boundary layer into the entire water column, where it can be transported long distances. Okay. If I haven't convinced you yet that small scale processes may be affecting intrusions, and I didn't tie that knot very well, I want to quickly show you a really nice recent paper by Ken Brink, who I think also may be online. Cross-Shelf Exchange, 2015, an annual reviews of marine science. So let me tell you his keywords here that turned out kind of small. Coastal physical oceanography, turbulent boundary layers, coastal instabilities, nutrient supplies, wind forcing. The first sentence of the abstract says, cross-shelf exchange dominates the pathways and rates by which nutrients, biota, and materials on the continental shelf are delivered and removed. The first sentence of the article says, cross-shelf exchange is arguably the central problem in coastal physical oceanography. So we have a problem. Some more nice quotes here. Cross-shelf exchanges deliver nutrients, govern residence times, transport planktonic life stages, enable sediment transport, remove contaminants. I won't read all these quotes, but I encourage you to go read this paper because it's very interesting. And the point being, to make progress, we need to be able to predict these things, not just pathways, but under different conditions. And we need to understand how the physical processes affect coastal ocean shelf ecosystems, which I think is what we're talking about in this. So the difficulty is we all have these assumptions we make, and Ken lists them as the flow field varies slowly with time. Non-linear effects are not important. Turbulent stresses and mixing are not all that important. And these are the assumptions of open ocean and frankly, open shelf physical oceanographers. Estuary people have different assumptions that are different. And the question is, so, and physical oceanographers want to tell you, the first thing they want to tell you is what scale they work at. Why is that? Why are we all sort of more or less in our own silos and uncomfortable moving to different? It's because of these things, the governing equations. I've listed only 4 of them. What they are is a nonlinear couple, difficult to find solutions to these equations. And what you actually have are 6 equations and 7 unknowns. So you've got to do something about eddy viscosity to make sense, to get your closure. So you've got some 7. And so what do? We try to simplify this problem. And so we have 2 tricks. And I thought, the physical oceanographers all know where I'm going here, I think. But to translate what the difficulty is, what is our problem? The first thing we do is non-dimensionalize. And so what you do is you select scales and you transfer everything in that equation to something non-dimensional. So, for example, the x and the y now become something with a length scale out in front. X prime, y prime. Temperature has a time scale out front. UV likewise. And I've just shown one example. So the UDT now has the scales are constant, so they come outside. They have this collection of scales out front. And now du prime dt prime is non-dimensional. So every term in that equation then ends up with a set of scales out front. They all have the same units, but they're all composed of different collections. They all have the same units because that's the way equations are. And so you can compare the magnitude of those different collections, and that's what we do to decide which of the terms we can leave out. So in the open ocean, continental shelf too for that matter, Coriolis is important. So we divide through everything by omega u. Those are our scales for the Coriolis term. And what you end up with some ratios that now have no units, and they're familiar. You'll recognize the names of them. The Rossby number tells you that you can leave out the du/dt term in a general room. The Rossby number, the Ekman number. The Reynolds number is just a ratio of the Rossby number to the Ekman number, scaled by the aspect ratio of the water depth to the length squared. So the point is, every one of us has our own selection of scales that we use to simplify this equation. And so we're not comfortable using other people's simplifications till we understand what they are. And so this is just a table from Cushman-Rosen, I think, just showing the length of potential scales you can use. They're not hardwired. You're supposed to use your experience and your intuition to come up with them properly. And they may not all apply to all the terms in the equation. That's the other problem. So our other trick is called gauge theory or asymptotic expansion. And what you do is separate u into a bunch of little u's, each with a small scale factor of increasing degrees out front. And then you plug that in. So for example, if you plug it in the u dz, you come up with a whole bunch of new terms. That doesn't look simplifying, does it? So we insist then that terms with like powers of epsilon out front balance each other. And so the order 0 equation is geostrophy. And then from that, you can add more and more detail depending on how far down the ladder you want to go. However, it's not true that scales don't affect other scales. You know the viscosity poem, right? Big worlds have lesser worlds that feed on their velocity. Lesser worlds have lesser still. And so on to viscosity. That's terms affecting other scales, frequency affecting other frequencies. And so our two primary tricks to simplify our equations need re-examination. And so I guess I just want to talk about the path forward briefly. I'm going to run over. I'm sorry, Bill. How are we going to make progress? I think we can count on our platforms, our sensors, our computer capabilities helping us with the straight up logistical problems. But I think we also desperately need interdisciplinary engagement. For example, that can help us select our length scales for any manner of things in ways that we as physical oceanographers did in grad school and never thought about again. This is a new way, I think, for us to think about what length scales we choose to emphasize. And it can also help us decide how much extra detail you need to add past geostrophy. We all need to make more effort to escape our silos by examining these scaling tricks that we use. And I think a good way to force us to do that is through this interdisciplinary engagement. We need to be willing to revisit these scale things as often as necessary and push our students to consider a range or perhaps wider range of scales than we did when we were starting out. And I think people like me, I'm an observations bigot. We need to be better at modeling. I haven't modeled since I was in grad school, but maintaining that capacity would have really helped me through my career. So I hope that this gives us some food for thought in terms of how we're going to engage with each other this week in terms of plotting our path forward. Thank you.
Thank you, Dana. We'll entertain anyone with questions. Please come forward.
I guess I should also mention that not just forward cascade, but there's also a reverse cascade where eddies, high frequency things influence lower frequency things, eddies influence the mean. Becoming more and more apparent. Ricard?
Thanks so much, Dana. You certainly gave us food for thought where you went from the scales as well as the intellectual scales, excuse me, from understanding what are the processes right from the micro scale through to the macro. I've got a couple of general comments. Firstly is that I think both with Federico's talk and yourself, you emphasized the role of tides on the shelf and their function for both biological as well as physical processes. And I think perhaps where we live off very much drier coasts, but we've ignored this important aspect of tide and freshwater discharge. So I think that's something we need to take into this afternoon. On a more specific level, though, I'm interested in how you said how it's difficult to scale up this carbon export problem. You know, we need to understand this because this is what we're, and especially with the role of salps and these sort of fast-growing gelatinous zooplankton, their contributions to the abyssal flux. So we're all kind of interested in canyons, and so my question to you is: do you know enough of the location and structure of these canyons, and are they foci for where you could or should maybe put in current meter moorings to examine this deep ocean flux, not the surface stuff that we all get worried about, but the deep ocean flux to the abyssal deep? Are those transport conduits both for maybe pushing stuff up, nutrients, but also taking carbon back down?
Yes, canyons. So in the South Atlantic Bight, we don't have canyons, except there is one on the map south of Cape Hatteras Canyon, but it's not there. It's just on the maps. So I think the responsible thing for me to do is direct those questions to John Klink, who's done quite a lot of work in canyons, and with exactly these kinds of questions in mind. So I'll encourage you to talk to John. How's that for ducking, John? Ricardo, did you have a question?
Oh, yeah. First question is, I'm not that familiar with that region, but you showed these intrusions of cold water in your first slide with high nutrients. So the question is, what is driving those intrusions? Is the Gulf Stream transport? Is the transit of a cyclonic eddy? Or is wind? And I have a second question. So, well,
Right, so it's a combination. The upwelling in the meander troughs is pretty well documented, and that's just a vorticity argument. But it's right up against the shelf, so it could spill onto the shelf if the wind forcing is right, and if it's going at a speed where that results in enough water arriving to be distributed. And it happens that is what happens in these regions of eddy decay. We don't know how often because we're not out there with a ship to measure it very often. We had an observatory out on the shelf, which was where that red dot with the progressive vector diagram originated. Several of those towers, Navy towers out there instrumented for a good while. And we saw in summer some intrusions, some arrivals of quite cold water. But we don't know what's in it because we didn't have.
but everything is related to eddies or is changes in the transport too?
Gulf Stream meander eddy upwelling is what, based on insufficient data, frankly, I mean, this work is 30 years old and we have not been out on our ships routinely enough to capture it over and over and discuss interannual variability. But the quantity of nutrients from that process is an order of magnitude better and higher than anything else. But it requires appropriate shelf stratification and upwelling winds to do.
Can I make it? It's fascinating that you showed two models. One, it was, I think that it was by Najar, and the other by Janko. One says that the South Atlantic Bight is a source of CO2 and the other is zinc. The question is, what's the difference between these models? I mean, what it makes such a big difference?
Air seed fluxes are poorly constrained. I think that's one good reason. Another is we don't know the incidence, how often we get these eddy inputs. I think the estuarine component is also poorly constrained. There continues to be argument about whether the marshes are a source or sink of nutrients to the shelf. And so we just have big error bars on every term. And so the choices you make when you're modeling determine your outcome by very slim margins. And I'll direct further questions to Bill, who's actually a chemist. I'm a pretender when I'm being interdisciplinary.
Yeah, Dana, thank you for the for the report. I have a few comment about your talk. You mentioned that the cross-shelf exchange mostly took place in the deep part or in the bottom Ackermann layer, hopefully because of the western boundary current rubbing on the continental slope, which is understood well. But have you thought about the other driving mechanism, like the cross-shelf penetrating front, and also the so-called arrested topography wave that the western boundary current set up a pressure head at the narrow shelf in the south, so that it kind of set up the streamlines spread over the shelf downstream. That could also generate significant cross-shelf exchange, perhaps in the surface, maybe not directly. That's obvious in the satellite image, but they have these dynamics and seems to can sustain for a long time. Have you compared or have you think about this kind of processes?
No, that's a good point. We have had an extremely good modeler associated with the southern end in the past, Cisco Werner, who used finite element modeling, and then Ryan Bland following on. But I think the narrowing shelf south of Georgia has gotten relatively little attention relative to what it needs. I mean, we've proposed several times to do things of that nature. And I can confess that I continue to struggle with the arrested topographic wave paper. So there's that problem. But certainly, if we had more data, we could consider more processes. And so that's a point well taken. Lisa.
Hey, Dana, that's a great talk. I really like the last slide in particular. I think there's some very fundamental things to think about in terms of how we tackle these issues moving forward. My point went on from Ricardo, really. So something I think about in relation to my work in the Agulhas current also is this issue of, with climate change, are the western boundary currents getting stronger in the sense that they're mean kinetic energy, and so the mechanism of Ekman veering, for example, may be amplifying over time. And is that kind of our primary nutrient supply to the shelf? Or the eddy kinetic energy of these systems, right? If the eddy kinetic energy is increasing, then you've actually got kind of maybe more porous front where you might get more exchanges closer to the surface, but you also get this upward pumping related to that meandering. So there's a lot to untangle here about, essentially, I guess what I think about is ultimately, do we expect the shelves are going to get cooler and have more nutrients over time? Or do we expect the opposite? Or do all these things going to balance out? I mean, that's kind of the million-dollar question for me. And I wondered what your thinking was along those lines.
So I guess I'll develop an opinion on your point about increased eddy kinetic energy being fed onto the shelf. And I think that will certainly change how these intrusions cook nutrients into organic matter. Because it will certainly disperse the nutrients faster. We think maybe we can use oxygen as a proxy for production, but not if it's being mixed away by mixing. And if it changes, the shore side into a leafy boundary, which it seems to be in the South Brazil because of this tidal mixing that Federico mentioned, that could certainly change, I think, what's going on. Without understanding the sequence through numerous intrusions, it's really hard to speculate. And you need to model this thing well. And I think we're getting there. And the Google work is just really impressive in terms of examining things that are of direct relevance to this process. And so I think we're almost there. And that does not even require a finite element approach. That's ROMS, which theoretically is just wildly available and possible to do whatever you want with. So I'm not sure I can answer that question. I think I'll duck that one too.
Well, let's thank our speakers once again. They are obviously going to be here for a couple of days for more individual questions. Pin them down whenever you find them. Thank you.